sponsors of this meeting for making this possible. Those include Jülich-Forschungszentrum, the Human Brain Project, HIBOL, which is the Helmholtz International Big Brain Analytics and Learning Laboratory, and Arabic Réseau de Bioimagerie du Québec, and the Neuro, of course, where this is virtually hosted today. So this is our second edition of the Gradients Workshop, but maybe this is your first time actually at the workshop, and maybe you're even new to this field of gradients. You're wondering, what are we talking about? So I can tell you to start off with a gradient in this context means a graded variation in a neuroanatomical feature. This could be within a region, or it could be across the whole brain. And they can also be identified with a lot of different modalities. You see some beautiful examples here from Julie Hudenberg's work of functional connectivity gradients and distance gradients as well. And even though we use many different methods and approaches here, what unites the field is an attempt to understand the spatial patterns of the brain and to better understand this intrinsic coordinate system of the brain. There's been so much research on this in the past year. There was even a special issue of Neuroimage dedicated to gradients. It had 36 articles by my last count. So it really shows it's a booming field. And today is this opportunity for us to get together and share our research and discuss it and collectively uncover more about how the brain is organized. Here we go. Okay, so there's 3 sessions today. The first session is going to be on bridging scales. This revolves around how local or microscopic variations relate to large-scale brain organization. We'll hear from Sukjun, myself, and Sean, and will be chaired by Thomas and Daniel. The next session will be on dynamics. This explores different approaches to modeling the functional dynamics of the brain. We'll hear from Katarina, Kayvan, Golia, and Mert, and the chairs will be Beth and Boris. And in the last session of the day, we'll look at Evo-Devo. This is where we have presentations on the conservation and divergence of gradients across different species and across the lifespan. We'll hear from Ludovico, Christine, Graham, and our chairs will be Sophie and Richard. So let's have a quick overview of what the day will look like and go through some logistics. So in a moment, I'm going to pass over to Thomas and Daniel, and they'll take care of us in the first session. Each session has 3 to 4 talks of 15 minutes. Some have been pre-recorded and some will be live. If there's a couple minutes to spare, then you'll have the chance to ask a quick specifying question, but please do keep broader questions for the end of the session, because we'll have a longer panel discussion of 45 minutes where you can ask questions that might be applicable to multiple speakers and can really bridge across that research theme. Immediately following the first session, we have gradients trivia, backed by popular demand. When the time comes, we'll send out a Google form so you can fill in your answers. It'll be a really quick little competition to see who knows their gradients. And then after that, we'll have time for a coffee break. So you can play trivia, then go grab a coffee. And after that, we'll have session 2, where we have 4 talks and a panel discussion once again. Then we have a longer break, time to go get lunch or dinner or go for a walk outside, and then a 3rd session. And I want to draw your attention to this last part here, where we have a social gathering and poster session on Wanda. Some people may have used Wanda already at other virtual conferences. It's kind of a stripped back version of some of these virtual worlds that you see for conferences, where it's just a place for us to kind of congregate into different virtual rooms. And there'll be a social gathering and a poster session. So we've already predefined poster rooms. We're doing it a little bit differently to a standard poster session. We've clustered people based on the themes of their posters into groups of like 2 to 4 posters. So you'll find that there's different poster rooms with a theme like individual variation or asymmetry. And you can go into that room, and would just recommend for the presenters to just take turns sharing their screen and going through their research to anyone else in the room. And so that will be both your fellow poster presenters and also onlookers. So hopefully we can invigorate some conversation, and it might even be like being in one of those corridors of posters at a conference again. So you get to know the people standing next to you as well. So that's the overview of the day. Then a little bit of housekeeping is you'll notice that all your microphones and videos are kind of slammed shut at the moment on you. So they'll remain throughout the talks, but in the panel session, we'll automatically change it so that everyone has the option to turn off their video and unmute themselves as well. But for the moment, you're all muted and your videos are turned off, just so there's no distractions during the content of the talks. While that's occurring, you're welcome to post any questions you have in the chat. So as I said, that each talk will have 15 minutes. If the talk goes for say 12 minutes, and maybe we'll have time for 1 or 2 questions, so you can post those questions in the chat, but then panel session will open it up a lot more. We're live streaming on Vimeo as well. So if your bandwidth doesn't support Zoom for some reason, or if you know someone who hasn't registered, feel free to go and follow on the live stream instead. And you can also ask questions even if you're not in the Zoom call via Twitter, and you can follow along with proceedings. So we're always posting at Gradients MTL, so you can follow along there as well. I think that's all there is from me, and I'm going to hand it off now to Thomas and Daniel.
Hi, everyone. I'm Thomas, and my co-host is Daniel.
Hi, Daniel. Very excited to be here.
So, should we just start, or?
Yeah, go for it.
Yeah.
Okay, sure. I'm gonna start. As Casey has already mentioned, there will be 3 speakers for today's session. The first speaker is Seok Jun Hong. He is from Songkyungkwon University. Correct me if I'm wrong, Seok Jun. In South Korea, but he has actually collaborated a lot with many people in this workshop, attending this workshop, including, of course, the host. So he will give a talk about mapping whole brain spatial temporal dynamics in autism spectrum disorder. So Seok Jun, I guess we will, do you need an alarm or something in like 12 minutes, or it's fine? Or just let you know in 15 minutes, or?
I think I heard that I can give a 50-minute talk, or I can even shorten the length of the talk. Yeah. Okay.
In that case, go for it then. Yeah, if you overshoot it, we'll let you know. Yeah.
I'm gonna do fast. Do you see my
For audience with question, you can just message in the chat.
Do you see my screen?
Yes.
Great. So, yes, I'm Seok Jun. Thank you for invitation, Boris and Casey and our organization committee member. Today what I'm going to talk about is how we can map the spatial temporal dynamics in functional brain network in autism spectrum disorder. I think I'm probably the only one who's going to talk about clinical samples here. So hopefully it's not so boring. So for those who may not be familiar with this developmental condition, autism spectrum disorder is characterized by many different behavior phenotypes, but as a representative features, they shows those list of phenotypes, including social interaction deficit, communication anomalies, restricted and repeated behaviors with narrow interest, as well as altered sensory sensitivity, as you see in these figures. And then if you see the list of these, you may realize that these phenotypes cover quite a wide range of different level of cognitive functions. For instance, these first 2 may be more related to high order cognitive function. These restricted repeated behaviors may be related to both sides, either high order cognition or low level sensory motor anomalies. The first one is more toward the low level sensory anomalies. So this is quite interesting phenomena in the field that a single entity with the umbrella term with autism still shows this multi-level of cognitive deficits, as leading groups also give their opinion on it, like Simon Baron-Cohen says in his paper, that the co-occurrence of autistic differences in both low-level perceptual behavior and high-level social cognitive processing is central puzzle of autism research. So this is an interesting question, but regardless of this, there has been already many different neurocognitive or biological theory in autism. As you see in the slide, there are many different theories, but particularly related to this co-occurrence of both low-level and high-level cognitive deficit in autism, I may consider more seriously these two weak central coherence and erroneous predictive coding theories. To understand what they are, you may have to understand this cortical hierarchy system. We all know that as a human being or even primate brains, we always constantly have to predict what we're going to encounter in the external world. And at the same time, we receive the flooding information of the sensory evidence, and we compare these two, make the prediction error, and propagate up to the higher hierarchy. So you see this constantly circulating neuronal information processing, which may be probably one of the core mechanisms how we can adapt ourselves to this rapidly changing environment. So you can easily imagine if this balance is broken, then many behaviors can be affected. In the context of autism, there has been a already big theory that maybe they are related to attenuate precision in prediction process. So they're basically uncertain what they're predicting, which caused this diminished influence of top-down modulation, which again result in this overweighting of local sensory information that may account for many different types of behaviors in autism. So as you see, this hierarchical system give a big hint to understand this system-level pathogenic mechanism in autism. So now, if I talk about more general aspect of this cortical hierarchy, we are all excited about these gradient approaches, because especially the gradient one shows this large-scale cortical hierarchy. Also the represented in the figure in Mesulam diagrams. This gradient one shows this from the low level to high order functional gradients. Not just the hierarchy, but turns out that the other dimensionality reduced component shows cognitively meaningful axis, cross modality gradient, or task negative to task positive gradients. As a counterpart, what is more interesting is not just statistic property of cortical hierarchy, but dynamic properties also seems to exist. For instance, this is what the result from what we call the quasi-periodic patterns. I think many of you probably know this technique. Briefly explaining, if you have the lengthy fMRI volume, you randomly select the template, and then you go through the entire length of the volume, calculating the sliding window correlation, and you pick up only those segments showing high correlation and merge them to update the template. You go this iterative process, performing exactly same analysis until the template is not going to be changed so much. And then if you unpack how this template look like, then this is what you can observe. If I play this video, then you can realize that the flow of this global fluctuation just follow the gradient of the human brain functional network in 1, 2, 3. So it's not just hierarchy, but other functional axis too. So it sort of give you special domain of dynamic properties in cortical hierarchy. But then there is also counterpart as a temporal domain in the dynamic properties. There are many different domain of evidence claiming that our human brain shows differential time scale across the brain. For instance, this one is monkey electrophysiological evidence. This one is computational modeling. These are all neuroimaging finding. And then basically, if you focus on this figure, this is from the Michael Cole and Ito study, saying that if you calculate intrinsic time scale from the resting state fMRI, those primary sensory areas shows a shorter time scale, whereas you go higher and higher in the high order areas, they have longer intrinsic time scales. And basically you calculate this temporal autocorrelation function and then measure this time scale of this function, and then you take this value as intrinsic time scale. This is how they calculate this ITS, intrinsic time scale. So this is temporal domain of dynamic properties. And then this complex pattern of spatial temporal dynamics seems to reflect the principle of large-scale cortical hierarchy stream. But what about in autism? How are these hierarchy-related dynamics affected in these developmental conditions? And I was so much excited that I'm going to perform this intrinsic timescale analysis or QPP analysis in autism. Then, guess what? What it turns out that whenever I came up with the idea, it means there's already a study is published. So I found this paper from Japanese group that. This is really beautiful study. It's published in eLife, where they calculate this intrinsic time scale in typically developing brain in autism. And this is just profile of the intrinsic time scale. And then when they statistically compare this time scale, they found a specific area showing a group difference. For instance, this post-central gyrus on occipital lobe, but also tendency of in high order areas, which again are associated to major symptom severity in autism. So it's quite interesting finding. But then I also found a little bit of caveat of this study that this study was based on the small sample size, like around 25 subjects. Even for the independent study, independent data that they used also has like 19 subjects. And then they mainly focus on high functioning of autism. Then what was the really pity was they never really say anything about this potential underlying mechanistic factors. So that was what I wanted to see from this study. And then again, the temporal domain is only aspect that they study, and they didn't really show spatial aspect of this dynamics, like in functional signal flow. So we conducted a recently we conducted one study. This is the PhD student in my lab that we took the large sample data set and like 306 individuals, and then analyzed resting state functional MRI. And we combined 3 different approaches: intrinsic time scale, quasi-periodic patterns, and computational modeling. So I'm going to show you one by one for the new finding, but bear in mind that this is still ongoing project. So there must be some findings that look quite incomplete. So, but then you can always give me feedbacks or comments for that. So this is intrinsic time scale that we calculated. As you see, both controls and autism shows generally normal looking patterns, like those somatosensory areas that have a shorter time scale, whereas if you go further to DMN or high order areas, they have longer time scales. But when you directly compare between the 2 groups, that we found specific areas that shows the group difference, this motor area and then visual area, sensory area in visual cortex and the frontal areas. And then it was quite interesting to see that we kind of replicated the Japanese group's findings that, for instance, this post-central gyrus and occipital lobe, and also tendency in the frontal lobes. We also further going to checking the region-focused hierarchical profile. So we picked up several sensory and frontal areas. And then we sort out this intrinsic time scale across this known hierarchy. As you see, they are not perfectly follow and they increase along the hierarchy, but more or less they show increasing patterns as you go further higher hierarchy. And then in sensory areas, again, within this area, they shows either a reduced time scale or longer time scale in frontal lobe. So it's also quite consistent finding. We also now check the spatial domain of a dynamic aspect using these quasi-periodic patterns. And then you can focus on this from the 14 second to 15 second, because the other extremes are either continuing from the past or next QPP patterns. So this is a one segment of QPP. And then as the Shella's recent study, as you see these patterns, it starts from this motor and then visuals, and then it goes to the DMN. So you can imagine how this functional flow is going on over the whole brain. And then when we compare between the two groups in terms of the activity strengths, then this early period where this sensory area activation time, the autism shows rather decreased activity strengths. But then as you go further to higher order areas, like in this middle time or later time, they show rather excessively larger activity strengths in QPP. We also check the time to peak to understand the spatial temporal dynamics. And it turns out that these sensory, the sensory areas shows rather longer time to make the peak response in autism, whereas in DMN, it shows rather shorter time to make the maximum response of the QPP in autisms. Finally, we performed this computational modeling. Again, as I put in the title, it is ongoing work. It turns out that this computational modeling has this devil in the details. So we really play around many things, checking the parameters now. But here, as a first model, we took the Domitas 2019 neuron model, where based on this structural connectivity, we built the neural math model having these few biophysical parameter, recurrent excitatory connectivity, excitatory to inhibitory connectivity. And then based on this computational model, we simulate the signal, neural signals and convert to the bolts, and then calculate the functional connectivity and iteratively comparing to actual experimental connectivity map to tweak the parameters to minimize the difference between experimental and simulation. We've initially wanted to make the individualized model, but turns out that because of this noise data, we could not really make the reliable performance, so that we took rather bootstrapping based subsample group level approaches where we perform 100 bootstraps. And these were the free parameters. I'm not gonna go to details because of short time, but we basically targeted this excitatory connectivity and excitatory inhibitory connectivity. We could achieve like a moderate correlation between experimental and simulated functional connectivity. And then based on these parameters, when we profile these biophysical parameters, then interestingly, those the low-level sensory areas shows or increased excitatory and low-level sensory areas. Whereas as you go further, it doesn't make the increase WEE, the excitatory, but in excitatory inhibitory connectivity was rather decreased. So in the end, when we calculate the ratio between this EE and EI across the whole brain, is constantly shows this increased excitatory components. We are trying to evaluate the relationship between these computational modeling parameters and intrinsic timescale by simulating the signals, but we haven't finished yet this analysis, so it's an ongoing project. So finally, to summarize, atypical cortical hierarchy may underlie various behavior phenotypes of ASD as a system level mechanism. The current study focused on spatial temporal dynamics related to this hierarchical system using multimodal, multi-method approaches like intrinsic timescale, QPP, and modeling approaches. In autism, intrinsic timescale was shorter in low-level sensory, but longer in high-order transmotor system. QPP was reduced, shows reduced brain signal flow in early period where the sensory area was maximally activated, but increased in the later time. Both DMN and frontoparietal network was maximally activated. So it's purely speculation, but globally increased excitation across the brain may be related to this atypical spatial temporal dynamics in ASD, which is the hypothesis that we have to check in later. So this is the end of my talk, and thank you for your attention.
Thank you, Seok-Jung. This is very interesting talk, very exciting work. I think we're running short on time. So does anyone have a quick question you want to ask? I think one of the audience member asked you whether you've already published, is this already published? I think you said that this is work in progress, right?
Yeah, no, it's far from the code.
One quick question I have is that can you go back to your slide of about the intrinsic time scale? I was kind of surprised to see that the like the dorsal tension seems to have a relatively long time scale, maybe even longer than prefrontal cortex. And I thought that, like, you know, the prefrontal regions should have perhaps longer intrinsic time scale. Yeah. Is this consistent with the other literature?
I think it's probably related to the reliability of the signal per se, and this is from a byte data set, which is 5 minutes. So we're really suffering from the not being fully able to replicate it. The other studies like Ito or Watanabe studies, where they show beautifully this hierarchical changing of the intrinsic time scale. But as you point out, there are some specific areas looking quite weird, but this is what we have now. And then we still check what kind of signal properties make this unexpected intrinsic time scale values. So
cool. I guess let us move on to the next speaker. If other people have questions about for Seokjun, we can do it at the end of this session. We actually have like 45 minutes of discussion time. Okay, so for the next speaker, we have Casey. Oh, I realized I don't know how to Pecola. Yes. She's currently in pardon?
Yeah, that was good, Pecola. I don't know how to pronounce it. You can pronounce it any way you like.
She's currently at Jülich, Germany, and she's of course one of the organizer of this workshop. So she does not need further introduction. So she's going to talk about the machine in the ghost, cytoarchitecture and wiring of the default mode network. Go for it, Casey.
Thanks, Thomas. Yeah, so I want to talk about the machine and the ghost, where the machine is the neuronal architecture and the ghost is that mystical default mode. Okay, let's get started. So I think it's quite interesting to start off by thinking about how the conceptualization of the default mode has changed over the past 20 years. It was first described as this task negative network, and you often read about it being an offline mode of the brain, but that was subsequently challenged by a lot of research showing it was enhanced activity during tasks that draw on memory, such as context-dependent decision-making or narrative comprehension or social cognition. So several contemporary theories start to postulate that the default mode is actually really important to balance integration of information from external and internal sources. So you see from this really lovely review by Yuri Hason's group that came out recently, the default mode in this intermediary position brings in a lot of extrinsic information and incorporates that with prior knowledge of the world from long-term memory. And it's thought that that enables the formation of a rich context-dependent model of a situation as it unfolds in time. So that's primarily based on task fMRI. But in the resting state literature, we tend to see the default mode conceptualized as the apex of a processing hierarchy. So for example, stepwise functional connectivity, if seated in primary sensory areas, it progresses towards the default mode and then achieves a new stable state. And also the principal axis of differentiation in resting state functional connectivity runs from primary sensory areas in the more purple colors all the way to the red default mode. So again, emphasizing the default mode as an extreme opposition to primary sensory areas. And my favorite analysis from this paper, though, is that the peaks of the gradient are maximally distant from primary sensory areas when you look at distance along the cortex. So it really shows this separation of the default mode from primary sensory input. So we really set out to test these hypotheses and fundamentally provide a richer neuroanatomical characterization of the default mode, which seems to be quite lacking in the literature up until now. Okay, so first, what is the default mode made of? We started by looking at the composition of the default mode with respect to cortical type. These were first defined by von Economo and recently reanalyzed by Garcia Cerveza et al. We were particularly interested in these types because they index the centrifugal hierarchy that was described by Meslin here. So here you can see that the different types based on cytoarchitecture, there's a few different cytoarchitectural features that change as well. The most prominent is the granularity. So that's the prominence of layer 4, and it disappears at agranular. So here we're going conicocortical, you laminate 3, 2, 1, dysgranular, agranular. I'll keep the order the same the whole time, so you can just think of cortical types by decreasing granularity. But there's other features of cytoarchitecture that change as well. And if we look at this in the schema of Meslin, theoretically, this is synonymous with going from primary sensory on the outer ring to unimodal, heteromodal, then paralimbic and limbic. So this provides a cytoarchitectural proxy for proximity to either the external world or internal milieu. So in the DMN, if we look at an overlap of these maps, we find that conicocortex, this kind of primary sensory type, is almost non-existent in the default mode. And this might just be due to like a mismatch of the group level atlases that we're using. I would expect this small proportion of conicocortex actually disappears if you had single subject maps, but that's a challenge for another day. But there's a decent representation of the other cortical types throughout the default mode though. And if we compare the proportions to the rest of the cortex, we see that the default mode has an over-representation of ulaminate 1. See that all the different types have different proportions overall, but this is over-represented in the default mode, which does fit with the typical idea of the default mode as heteromodal. But I think this makes us rethink how networks are composed a little bit, because it shows that there's a wide range of cytoarchitectural types nested within the default mode. And the cytoarchitectural variability is more nuanced than cortical types as well. So we map cytoarchitectural variation within the default mode by measuring intercortical variations in cell body staining from big brain. Big brain is a high resolution volumetric reconstruction of an entire post-mortem human brain. We took a total of 62,000 staining intensity profiles. These are oriented in the direction of cortical columns. They run from keel to white matter, and they index variations in cellular density and soma size as you increase depth within the cortex. So you see these blue lines have very high density in these deeper layers, whereas the red lines are more flat. And these are all taken from within the default mode. We correlate these profiles to calculate microstructure profile covariance, and then we applied diffusion map embedding to extract the principal axis of variation in cytoarchitecture. This is the first eigenvector. It explained 42% of variance approximately in the default mode, and it illustrates what we've come to think of as an outside-in pattern of cytoarchitectural differentiation. So we call it outside in because you notice there's a lot of local gradients that run towards the limbus. Let's have a look at the medial parietal area, for example. It goes from blue to red, from posterior to anterior. But if we flip it and we look at the frontal cortex, we actually see that the gradient goes from anterior to posterior. So the increases seem to be going inwards. You can also see this really nicely in parahippocampal gyrus, that the gradient value increases as you move in towards the hippocampus. And so this outside in refers to outside in towards the limbus. And the limbus here is defined as the edge between the cerebral hemispheres and the diencephalon. So the outside in gradient is distinct in how it represents association cortex relative to the sensory-fugal hierarchy. So here we have the gradient values for each type within the default mode. So if we expected that the outside in gradient is parallel to sensory-fugal hierarchy of these cortical types, we'd expect a diagonal here. But we do get similar anchors. It seems that the sensory-fugal hierarchy extends all the way from conicocortex to agranular, whereas default mode is a little bit more restricted, but it abuts to those ends as well. But it has a very different representation of ulaminate, 321, and dysgranular. So I think it's important to point out that these two gradients are not parallel, but they're also not orthogonal. Okay, then, so overall, just from this cytoarchitectural picture, we've learned that the DMN is heterogeneous and it's organized along an outside-in gradient. Relative to other networks, we also find that many other networks, they have a variety of cortical types as well. So this is just a quick overview of the fact that all the networks have multiple cortical types in them. The default mode has a distinct distribution, though. It's most similar to ventral attention network. If we look at big brain, though, where we can really evaluate heterogeneity, we see that the default mode does have the highest level of cytoarchitectural heterogeneity compared to the other functional networks, although closely followed by a ventral attention network as well. So maybe something funny is going on in there. So next, we want to keep building up this neuroanatomical characterization of the default mode in terms of its intrinsic architecture, as well as regional differences in its extrinsic connectivity. We already know that the default mode occupies this unique position in the cortical mantle. So we want to ask how the topography influences regional differences in connectivity of the default mode to the rest of the brain. To do so, we combine diffusion-based tractography with distance to calculate navigation efficiency between 400 parcels across the cortex. I think not only does navigation efficiency elegantly fuse distance and tractography, but there's also research showing that it closely approximates invasive track tracing connectomes in non-human primates. So it seems to be well suited to map low-cost wiring in the brain. Then returning to our question of communication of the default mode to external versus internal sources, so whether it's more predisposed to communicate with sensory areas or more limbic areas, we look at navigation efficiency to each cortical type. And if you look at the other networks, you see there's an imbalance or some preferences in which cortical types they have stronger navigation efficiency to. Limbic is quite obvious, very high navigation efficiency relative to dysgranular areas compared to other cortical types. But the default mode has this type of balance that we don't see in other networks. And compared to null models where we set up a homogeneous balance, we find that only the default mode really matches this null model in terms of its balance in communication to all these different cortical types. So it seems that there's a potential of the default mode to equally communicate across the sensory fugal hierarchy. When we tested whether cytoarchitectural gradient contributes to this balance, the average navigation efficiency decreases along the gradient. So the outside aspect of the default mode has higher navigation efficiency to everywhere on average, whereas the inside aspect has lower navigation efficiency. It's closer to the limbus. It seems a little bit more insulated. And this effect was especially driven by more granular types. So the outside aspect has especially heightened navigation efficiency to more externally focused areas. And in essence, this shows that extrinsic connectivity of the default mode is also organized along the outside-in gradient. Okay, so the structural manifold of the DMN demonstrates the potential for equipoise communication across the sensory fugal hierarchy, but understanding how this potential is actualized really requires an examination of ongoing fluctuations in brain activity. So to do so, we modeled the effective connectivity using resting state fMRI and regression dynamic causal modeling. So RDCM is a highly scalable generative model of effective connectivity. It was developed by collaborator Stefan Fressel, and it lets us inspect the directed signal flow throughout the entire brain. So first we look at the type effect again, and we find this balance is present in the efferent connectivity, so DMN sending information to the rest of the brain, but not so in afferent connectivity, where there are preferences to certain types in terms of the incoming information of the default mode. So we see that the DMN can broadcast widely across the sensory fugal hierarchy. And then we look at the gradient effect, and we find that average afferent connectivity decreases from outside in, but we don't see such an association for efferent connectivity. So negative correlation in the afferent connectivity, it mirrors what we saw in the navigation efficiency. So again, showing that this inside aspect of the default mode seems to be relatively insulated from input. Also, unlike the navigation efficiency though, we did not observe a type by gradient interaction for the afferent connectivity. So combining the fMRI-derived afferent connectivity results with the navigation efficiency results, we would hypothesize that the outside aspect receives the majority of input of the DMN from many different cortical types, but based on this navigation efficiency, it seems that more granular types may have higher fidelity or faster communication to the default mode. So I think our description of the DMN's neuronal architecture sets us up to try to formulate a theory on what information enters the default mode and what the default mode does with that information. So input for the to the default mode is organized along this outside in gradient. So let me orient you a little bit here. We're going to put everything that's not default mode into this little bar plot. And then the default mode is represented by different points, the different regions in the default mode. And it's organized by outside ingredient and then navigation efficiency to primary sensory areas. So something like the insulation of an area as well. So we see that if we look at the top 5% of afferent connectivity, it comes from a few different cortical types, but it really converges largely upon a subset of the default mode, leaving this section relatively insulated from input, as I was mentioning before. I think this zone of convergence is quite interesting. It definitely fits with this idea of the default mode having many hub regions as well. And I think the convergence, because it's collecting information from across the sensory-fugal hierarchy, it can allow for this unique recombination of information that then kind of lights the fire of communication within the default mode. Because we know that there's really dense communication in the default mode based on functional co-activations, as well as a long-range structural connectivity that's costly. It's being used in the default mode. And so the variety of cytal architecture in the default mode tells us that as information is moving through the default mode, it has to constantly be transformed. And I think it would change dimensionality and it's reconstructed. So we put forward that the default mode reconstructs information in a distinctive manner that's not enabled by the directed flow along the sensory fugal processing streams. And then finally, we see that the default mode really widely broadcasts this information back across the sensory fugal hierarchy, speaking to its potential to shape our predictions and expectations at many different levels. Okay, with that, I'll wrap up and say thank you, thank you to everyone who's supported this project, but especially Margie Garber, who started this project when she was rotating through the lab as a PhD student, and Stefan for always being helpful with anything DCM related, and of course Boris, because we've been having a lot of fun discussing everything to do with the default mode. And if you're interested in combining cytal architecture and histology with neuroimaging, I'll direct you to Big Brain Warp as well, where we have a lot of code and data and tutorials to help you do projects like this. Thanks.
Thanks for this excellent talk, Casey. Very intriguing. I think we have we have time for a question, maybe a question or two. Anyone? If not, I can ask my own question. So, Casey, in this study, you're actually relating the DMN to the big brain like cytoarchitecture, right? I'm wondering if you have also, now that you're at ULICH, have you also looked at their other cytoarchitectonic atlases with yeah, and how does that relate?
Yeah, I haven't looked at the other cytoarchitectonic atlases yet. One problem at the moment is being able to do transformations to any of the other brains, for example. And so in terms of moving to individual brains, it's still quite a challenge, but there are like the next generations of big brain are being sliced and scanned at the moment. So I think it's kind of a future project, but relating it to previously constructed brains, the transformations just haven't been fully worked out yet. In terms of, for example, the ULICH Brain Atlas, which was just published earlier this year, I believe, at the moment that is really well constructed for many regions, but not for the regions of the default mode. There's a huge gap map for the frontal cortex, for example. So I think that the established cytoarchitectural areas sometimes aren't well characterized enough for the areas that are very predominant in the default mode. But I think that looking at individual variability is something I'd love to do, and hopefully we could get there with quantitative T1 as well.
I realize that two people have raised their hands. Why don't we just answer the two people's question and then we move on?
I'll try and be quick.
Yeah, Nicola, do you want to go ahead?
She may not be able to.
Oh, she needs to unmute. We need okay. Let's see, us to unmute.
Thank you. Now I'm on mute.
Nicola.
Thank you very much.
Yes.
Lovely talk, Casey. Really, really interesting. I've got a question concerning how you identified the areas in the big brain. So, did you personally?
It's just, yeah, no, it's just based on transformation of the group average functional networks onto big brain. So there's not really any specialization for big brain. I think that's why there might be some misalignment, I think, still in terms of like what would the default mode really look like in big brain. We don't.
Right, and also you don't know exactly how many cytoarchitectonically distinct areas are located within each one of the default mode hubs, or right?
No, not fully. I haven't really investigated much further because of the huge gap maps that I know are going to be there for frontal cortex. But I think in other areas like the parietal areas, we could definitely test that out.
Yeah, no, it's just because, for example, the one that you showed, on the medial surface, the posterior cingulate cortex, you've got a lovely gradient from posterior to anterior, which mostly or covers a lot of Brodmann's area 23 or autonomous area LC, and that change in that gradient that you see it reflects differences in cytoarchitecture. So so the part that, for example, that is red in your gradient, that would be most probably dysgranular portion of Brodmann's area 23.
Yeah, it's nice validation to know that as well, that even though this is a purely data-driven method, that we do see quite a good alignment with what our expectations would be based on the areas as well. Even if it is like data-driven ingredients, we see that there's suddenly a sharper change exactly where we'd expect that shift to LC. Yeah, it's definitely
it's just because it's an area that I'm mapping at the moment, so that's why it drew my attention. Okay, thank you very much.
No, thanks, Nicola.
Okay.
I can maybe ask, Casey, beautiful talk, and the work is amazing, obviously. I just have like one question for the heterogeneity in the DMN with respect to cytoarchitecture while you were presenting this.
Yeah.
Would you expect that, I mean, this is like I think like in some of the previous questions, it also came up a little bit. Would you expect that inter-subject variability plays a role also for, say, if you take something that's like functionally really consistent across subjects, would you then expect less cytoarchitecture variability or the same amount? Or is there like a way to quantify this?
Well, maybe I'm not quite going to answer the question, but I think when we're looking at the level of networks, we should always expect heterogeneity. Like, I think it should be a defining feature of a network that there has to be variation in cytoarchitecture. So I don't think the heterogeneity would go away if we refined the map, for example. But just, I don't think I fully answered your question. Maybe.
I mean, that's maybe also like a question to the functional folks out there, whether, you know, we use the EU map of the DMN, but there's possibly like a specifier that is kind of like a probabilistic version of it, like how robust it is across subjects, and then what could just vary this across individuals, the DMN definition, and see whether this heterogeneity relationship with the cytoarchitecture still holds.
So Ruby has done some work on estimating these networks in individual subjects. And as a result, she also maps, for example, how variables are the different networks. So in general, we find that the sensory motor networks are less spatially variable across subjects, whereas the association networks are more variable. I actually cannot remember for the default network whether it's in between or is actually the most variable. So yeah, but I guess Boris, I think what you're saying is that if you have this variability across subjects, so when Casey map it to the cytoarchitecture, like there'll be greater misalignment for these networks that are more spatially variable. Is that right?
Yeah, or in other words, whether we can have something that's like 100% DMN in all subjects, 90% DMN in all subjects, 80% DMN in all subjects, you know, that we kind of probabilistically specify where the DMN is situated and then just use maybe a more conservative definition of the DMN.
It sounds like we could using Ruby, like if we
Mm-hmm.
have many different networks, we could be like, well, imagine if the big brain has this, was this person.
Yeah.
And run it like that as well.
Right.
Yeah, I think the individual
okay.
variability is difficult to get around with big brain at the moment. But
so Ruby has already put those networks up. So if y'all want, you can easily just download them and average it yourself to get this probability map. Yeah. So if you have any issues, just email me. Yeah. Just email us. Yeah.
Cool. Thanks.
So Daniel, you want to do the last speaker?
Let's do it. Yeah. Thanks, Thomas. Casey, beautiful work as always. It's been a real treat. And to wrap up the session on scales and gradients, we have a talk by Sean Frodist-Walsh, who's currently at New York University, working as a postdoc in Xiaojing Wang's lab on dynamical models primarily. And he comes from a background investigating neuromodulators and networks involved in memory in humans and non-human primates. And we'll be giving a talk today on gradients of receptor expression in the macaque cortex. We're really excited to hear about it. So Sean, take it away.
I don't hear any sound myself.
Sasha, could you unmute the recording, please?
Hi, I'm Sean.
There we go.
And I'm a postdoc in Xiaojing Wang's lab in New York University. Today I'm going to speak to you about our recent work, Gradients of Receptor Expression in the Macaque Cortex, that is part of an ongoing collaboration with Nicola Palomero-Gallagher and her wonderful anatomy group in Jülich in Germany, as well as with Ting Xu and Daniel Margulies. The mapping of the connectome across species is one of the major recent achievements of neuroscience. However, connectivity alone is insufficient to explain neurocircuit dynamics underlying brain functions, as these also depend on the neurotransmitters and receptors used for communication between neurons. Bearing this in mind, Nicola and her team in Jülich used in vitro receptor autoradiography to measure the density of 14 receptor types across 109 areas of the macaque cortex. We aim to analyze the principal patterns of receptor variation across the cortex and to understand them in relationship to the large-scale anatomical and functional organization of the cortex. After they had measured the density of these receptors across many different slices, we mapped their in vitro data onto the cortical surface. This allowed us to project the densities of 3 types of glutamatergic receptors, 3 types of inhibitory GABAergic receptors, and 8 types of neuromodulatory receptors onto the cortex. Now, this is quite a lot of information, so to simplify the complexity somewhat, we performed a principal components analysis, or PCA. The first principal component, shown here, which we call the principal receptor gradient, accounted for most of the variance in the receptor data, with the top 5 components accounting for 95%. We can then project each cortical area into a 2D space defined by the first 2 principal components, which we call the receptor space. We can recognize that anatomical and perhaps functionally related areas, such as the visual areas here, tend to cluster together in receptor space, indicating that they tend to have a similar receptor expression. Let's now look at receptor expression in different cortical areas along PC1. Here we show the receptor fingerprint in area V1, where a higher receptor density corresponds to the colored bar being closer to the edge. As we pick areas that are progressively further along the first principal component, we see that the expression of most receptors increases. This led us to believe that perhaps the first principal component was simply encoding the total number of receptors per neuron. Indeed, we saw that the principal receptor gradient explained 99% of the variance in the total receptor density across the cortex. We also see that areas near the top of the receptor gradient express about 4 times as many receptors per neuron as areas near the bottom. So receptor density per neuron increases fourfold along the principal gradient. We then look to compare the receptor expression to other anatomical. First, we analyzed the retrograde track tracing data of Henry Kennedy and colleagues. We estimated the cortical hierarchy by examining the pattern of laminar connections between pairs of regions across the cortex. The hierarchy, estimated in this way, was strongly positively correlated with the receptor gradient. Putting this together with this previous information suggests that areas that are low in the hierarchy, such as neurons and sensory areas, have relatively few receptors. However, how might neurons that are higher in the hierarchy be able to hold more receptors? To examine this, we collated data from a series of studies by Guy Elston and colleagues, where they measured the area of the dendritic tree. We found that the size of the dendritic tree of basal dendrites and pyramidal neurons increased systematically along the principal receptor gradient. So neurons in areas near the top of the hierarchy may grow large dendrites, which may allow them to house more receptors. Myelin has been shown to inhibit the creation of new synaptic connections. The ratio of the T1 weighted to the T2 weighted scan has been proposed, not without controversy, as a potential marker of cortical myelin. We found a strong negative correlation between the receptor gradient and the T1-T2 signal. We decided to investigate the relationship between receptor density and myelin further by looking across layers within an in vitro slice of V1. Here we see that myelin peaks in layer 6B deep in the cortex and has a second peak in layer 4B, and it's also generally higher in the deep layers compared to the superficial layers. Although there is some variation across the receptors, generally they tend to follow an opposite pattern, with dips in layer 6B and 4B, and generally higher receptor expression in the superficial compared to the deep layers. So our analysis suggests an inverse relationship between the receptor density and myelin across cortical layers, and perhaps also cortical areas. Ting Xu recently built on Daniel Margulies' discovery of gradients of functional connectivity in the cortex, and she and her colleagues defined joint gradients of functional connectivity across the human and macaque cortex. Here we found that the first two gradients of functional connectivity were significantly correlated with the primary receptor gradient. This suggests that variation in receptor expression may also shape in vivo interactions between cortical areas. We then used Ting Xu's cross-species functional alignment technique to map classical resting state networks from the human onto the macaque cortex. Here, if we look at the first principal component of the receptor data, we see that the sensory networks and the higher cognitive networks are clearly separated along this principal component, such that the visual and the motor network has significantly lower receptor density than the four higher cognitive networks. Our analysis so far has been focused on the first principal component, which is a marker of the total receptor density per neuron. But we were also interested in differences in the patterns of receptors expressed across cortical areas. Here we see that the second principal component is mostly driven by differences in the expression of the serotonin 5-HT1A receptor. This component also clearly separates canonical resting state networks, with very low 5-HT1A expression in the dorsal attention network and high expression in the default mode network and the salience network. So distinct serotonin receptor expression between higher cognitive networks drives the second principal component. To investigate the relationship between our results in the macaque brain and those in the human brain, we first mapped the gene expression for the 5-HT1A receptor from the Allen Human Brain Atlas onto the human cortex. Then, using Ting's cross-species functional alignment technique, we translated this onto the macaque cortex. This allowed us to compare it with 5-HT1A receptor expression, and despite the differences in techniques and species, there was a strong positive correlation between human 5-HT1A gene expression and macaque 5-HT1A receptor expression. Furthermore, the human gene expression and macaque receptor expression mapped onto resting state networks in an extremely similar way. Using receptor autoradiography, we also found that there was a strong relationship between the macaque expression and human receptor expression across cortical areas, and also to a lesser extent with the rat. For the last two slides, I'm going to give you a little hint of related work that we've been doing. In this work, with the research work called Elise Klautzman, we examined the NMDA and AMPA receptor expression across the cortex. Given that AMPA is much faster than NMDA, it's usually thought that AMPA receptors should dominate in the sensory cortex, whereas NMDA receptors, which are thought to contribute to working memory, should dominate in higher hierarchical areas. However, when we analyzed the macaque data, we found that the fraction of NMDA receptors actually decreases along the cortical hierarchy. Now, similar result has been found recently in the human cortex by Nicola Palomero-Gallagher and Alex Goulas and colleagues. We then built a large-scale model of the cortex that matched these NMDA and AMPA ratios. This was driven by long-range feedback connections, mostly being onto NMDA receptors, and long-range feed-forward connections being mostly targeting AMPA receptors. We found that this somewhat counterintuitive NMDA-AMPA gradient was actually critical for realistic fast propagation of sensory information from sensory areas to the prefrontal cortex in a time that matches the signatures of ignition in experiments of access of stimuli to consciousness. Whereas models that had the NMDA and feed-forward receptors took far too long to propagate the stimuli to the cortex. In another recent modeling work, we examined how dopamine D1 receptors and long-range connectivity determine distributed working memory activity and performance. We showed that lesions to regions with a greater D1 receptor density cause a greater disruption of working memory activity. And this model replicates many findings from the working memory literature, such as the distributed pattern of activity across the cortex, the inverted U response to dopamine, and increased resistance to distraction with dopamine stimulation. So to conclude, we found a principal gradient of receptor expression that increases along the cortical hierarchy. Brain regions with more receptors also tend to contain neurons with larger dendrites and less myelin. We also found that higher cognitive networks are distinguished by the pattern of serotonin receptor expression. So with that, I want to say thank you very much for listening, and also thank collaborators and funders. And thanks very much to the organizers of this workshop. Hi.
Fantastic. Thanks so much, Sean. Are you there? Yeah.
Hey, yeah, sorry, I am here. I am actually in a Florida hotel right now, which is why I pre-recorded the talk. So if my connection drops, well, I guess we'll just do our best. But I'm happy to take any questions now quickly if there are, or else maybe we can go straight into the group discussion.
Fantastic. Are there any questions for Sean? Please. Great. Oh, Sukshan.
Yeah, Sean, thank you so much for a great talk. This is a beautiful study. There are many shocking results for me. So there could be many questions, but if I pick up one question, when you show us the clear, you clearly distinguish between the salience and default mode versus the others, right? To me, it reminds me that what is the role of this salience network? So because we tend to believe that this default mode network is really far from the external world, and they try to internal mode. And then what do you think? What do you have as for the opinion that what is the role of the salience? Whether this is similar to, is there any function similar to default mode network, or yeah?
Yeah, so this is something that we're kind of starting to explore now, maybe with a, like a large scale dynamical model, and trying to think about why do we see this pattern of serotonin 5-HT1A receptor expression there. And this is mostly tends to have an inhibitory effect. And we actually kind of see, if anything, an opposite serotonin receptor expression pattern in the dorsal attention network, where we see greater predominance of the 5-HT2 serotonin receptor, which is more kind of excitatory. So what I think now is that perhaps when you get serotonin expression, it might serve to actually inhibit the default mode network. And if you get a lot so that it excites it engages the 5-HT2 receptors, you might also excite the dorsal attention network. And the way I'm kind of thinking about this is that these two large-scale networks are maybe kind of in competition with each other, and that serotonin kind of release can kind of just tip the balance perhaps in favor of the dorsal attention network. And I think that's kind of consistent with some results that serotonin release is happens in kind of surprising situations. Now, what the salience network itself does in that situation is something that I really want to explore in this large scale model that we're kind of not there yet, but I think it might just kind of help bias this competition between these two other large scale networks. But of course, we have a lot of experts on networks here today, so I'd love to hear other people's ideas too. So I can steal them. No, yeah.
Excellent, excellent. Other questions? I think at this point the panel discussion has naturally begun. So feel free to jump in.
Yeah,
Sean,
go
ahead.
Okay. There's some questions in the chat. Daniel is asking, still thinking about layer dependency. It is to my understanding that, oh, sorry. So just now while you were playing a video, I don't know if you saw the question whereby someone was asking you how you handle the fact that there's layer dependency in the gene in the receptor expression. Yeah.
Right. Yeah. So I think maybe Nicola briefly touched on this. So I think as you'll understand, right. So this is 14 receptors across 109 regions, and then we were trying to link it with fMRI data and myelin data and so on. So at the beginning, we're kind of ignoring the layer information for the moment, but this is not our plan. We want to really integrate this both in the analysis and the modeling going forward. We've already done some preliminary layer dependent modeling, but this is really work that I think is going to be developed mature more in the next couple of years. So there's a postdoc working with Nicola Palamero-Gallagher called Thomas Funk, who's really working on the 3D reconstruction of the receptor, all of the receptor data in the human and the macaque. And once I guess his work is finished, we're going to have a lot more flexibility in terms of analyzing the layer dependence and many other analyses. So I agree that it's important. And I would say that we're just kind of not quite there yet in terms of even extracting all of the layer data for all the receptors, I think. Yeah, thanks.
Cool. Yeah, so let's see. Alexis is asking, could you please point me to the data set you used to perform alignment between humans and primates? Oh, by the way, if people want to just ask a question instead of just messaging, feel free to raise your hand and we can unmute you. Raise your hand in the Zoom way, not unmute. Your videos are off, so we cannot see you raise your hand. We cannot see you raise your physical hands. Yeah.
So I don't know. Sorry. Did you want me to answer, Tom?
Oh, sure. Point me to the data set you used to perform alignment between humans and primates. Yeah,
right.
That's what.
So this is using, I don't know if Ting is here, but Ting Shu's really wonderful cross-species functional alignment technique. And she used I think principally the Human Connectome project data and open data from the Prime Dre non-human primate neuroimaging collaboration. And I'd really recommend anybody who's interested. I know there's a lot of expert neuroimagers here, but maybe not as many people have worked with non-human primate data. And you know, we have had a very small role, but people like Daniel have been some of the leaders in creating this open data set of non-human primate imaging data. So I'd really recommend going and playing around with that. Now, I think another question in the chat was about, was this anesthetized or awake data? And there is both anesthetized and awake data in this open Prime Dre data set. And Ting has done this analysis with both anesthetized and awake data, and you can check out her paper with lots of collaborators, probably many of you who are here in your image in 2020. I think it's a really lovely paper that you can dig into the details on.
Cool. I guess the questions have all been answered in the chat. Does anyone else want to raise up any points or anything?
Can I ask a question to Casey?
Go for it. Yeah.
Yeah.
So your talk was also lovely, really like fantastic study. I was just wondering whether you know the recent study from the Gustav Deco, where he checked the kind of hypothesis about global workspace theory, which is related to consciousness access. When I saw your talk, I wonder those within this DMN, those area that is really inside, is it much closer to conscious access or more important to make the conscious access or not? So how Gustav Deco checked this hypothesis was calculating rich club connectivity for the functional connectivity, not the structural connectivity. And then it turns out that those rich club areas are related to some conscious access. The paper is published in Nature Human Behavior, and then I feel that you may want to check this and within this DMN, which area is more important to this conscious access.
Yeah, I kind of forgot that I read that paper actually. As soon as you said global workspace, it made me think like whether is it that the areas of the default mode that are receiving all this information are having conscious access, or the ones that are more removed. And I don't know if I really have ever worked out how to judge what conscious access is though. So yeah, like it may be related to your functional rich club, but I don't see how that's a leap to conscious access. What I do know is that the outside part of the default mode, so the areas that are more on the border and like away from the limbus, are the ones that are receiving the most information. But then it's also different to the ones that are sending information. So,
okay.
So the functional rich club, I would assume, is something like which areas like echo the most other areas.
You know, the paper, basically the message of the paper was the moment that we feel this consciousness access is the moment that one of the rich club nodes to start to propagate the information to other rich club nodes.
Okay.
So when I listen to your talk, whether those outside of default mode network play a role of this propagating the information across different rich club or inside. So.
So the communication within the default mode was quite odd, actually. I didn't show any of those results, but the connectivity, both based on like the navigation efficiency and afferent and efferent connectivity, didn't follow either the outside ingredient or the structural model within the default mode. So I would have to kind of use another data-driven method, I think, to understand communication within the default mode, because it didn't fit with the two more hypothesis-driven ideas that we had, either that that it's going along centrifugal hierarchy once it gets into the default mode. We don't find evidence for that, like it skips all over the place. And we also don't find that it's organized by the outside in. I think there's a different form of connectivity within the default mode that isn't revealed by the two axes that I was showing actually.
So I think this is a really interesting kind of line of questioning, Sukjun. And although my intention when I started in neuroscience was always to avoid consciousness, unfortunately I've been dragged in somehow. And we're kind of working on a model, like a large-scale dynamical model, kind of based on the connectome of access to consciousness. Well, I mentioned it in the slide in my talk with a really great young researcher called Elise Klatzmann and Xiaojing Wang and Stanislas Dehaene. And actually we're thinking not so much of the moment of access to consciousness as when it reaches the default mode network, but maybe more like when it reaches something like what we call like the frontal parietal network or central executive network and so on. So that's just how we're thinking of it. I think another thing to take into account is that, at least in that theory, it doesn't, when you go down to the level of neurons, it doesn't have to be a stationary set of neurons. It just has to be this large scale distributed pattern of activity that can integrate information or share information across large parts of the brain. So it could be the case that, you know, it's initially or mostly involved in communication, say from sensory areas through the dorsal attention network to frontal parietal or central executive network. But maybe in other occasions there's more of a contribution of the default mode network. That's how I think I'm thinking of it, at least.
Sophie.
Okay.
Oh, go ahead.
Yeah, I have maybe like one question that's like also to Sukjun initially, but then maybe also like a little bit more general about the session. Sukjun, in your talk, you showed like these beautiful biophysical simulation associations, but you mentioned that, you know, you work mainly at the group level and it was challenging to make them work at the individual subject level. And I'm sure like the people who do the simulations, they get these questions like all the time. What is like a good way to make these models work at single subject level? And then also, do we generally need to build biophysical models to actually be able to bridge across scales, or is like just a spin test correlation sufficient to do this?
That is like hardest question, which I'm not sure that I can answer for. But I can answer with respect to my experience for the individual modeling. First of all, my experience was I never really found a stable model optimization result whenever I work on the individual. So depending on the slightly different parameter set, your model signals is fluctuating here and there, so that in terms of stability is not so great when you work on the individuals. So I don't know. I cannot really answer what is the really best way to do this individual. Maybe Thomas has the answer for this.
Yeah, actually, I'm curious. When you say that you are having trouble, like what do you mean? So you are saying that if you run the model, when you run the Dermitas model twice, his optimization twice, you get two different results, or what?
Yeah. So when I set up a certain parameter for the model and then get the result, and then I have some sense how I should change the parameter so that I reach to my goal. But then if I change slightly some value, the parameter, like excited, like some values, then the model dynamics is completely different. So that I cannot really.
Yeah. So first of all, these kind of models are very nonlinear. So so a slight change in the model parameters could lead to a big change in the dynamics. So my question is that when you say that you have a sense of how to change the parameter values, I thought the Dermitas papers they do this by Bayesian optimization approach, right? You're tuning the model manually, or you're actually using the optimization?
There are some free parameters that automatically should be optimized, but there are also several hyperparameters that I set up. Like
as usual, there are some special parameters that are.
Yeah, but you recently published in bioRxiv paper that you now use two objective function. One is like functional gradient, and the other I don't clearly remember. But when I take object function or the other object function, the dynamics is completely different, so that I can really summarize it. Yeah. So
the other thing is that there's definitely a degeneracy in the model, in some of the model parameters. So you might need to constrain this. We can talk further. I feel like we're going too much in details now, but you might want to constrain the sign of some of the parameters. Yeah. Because I mean, John told me as well that that he didn't really harp on it in his paper, but in the Dermitas paper, but there is some degeneracy in some of the parameters. So like a positive value, if you flip it to a negative value, it might work just as well. Yeah. So that could also add to your issues. Yeah, we can have a chat further. Yeah. Or you can ask John.
So I think that a lot of the focus in the large scale modeling has been on trying to explain resting state functional connectivity patterns, and that's great, right? But maybe you might be able to figure out a solution if you also try to explain other things as well as resting state functional connectivity, right? So what might be, you might get an equally good solution if you have very strong recurrent excitation in the sensory areas as if you have very strong recurrent excitation in the prefrontal areas, for example. But those two solutions might not be as good as each other if you're trying to explain working memory or some attention task or some other things. So, you know, I think it's worth trying to also think about trying to explain different things with the same model. So for example, Sukchun, you have this really cool data on intrinsic timescales, right? I mean, are you able to, instead of just trying to explain the resting state functional connectivity patterns, also explain the intrinsic timescales with the same model, right? That might give a solution. And actually, I might bring this back to a question for Sukchun, because, you know, initially a paper from our lab that I wasn't involved in, from Rishi Chowdhury back in Neuron in 2015, they came up with a model that explained this variation in intrinsic timescales, and they actually suggested that the increase in intrinsic timescales, now this is meant to be neural activity, not bold activity, so there could be some difference there, but they suggested that this increase in intrinsic timescales was associated with an increase in recurrent activity along the cortical hierarchy. Now, if I remember correctly, you found a longer intrinsic timescale in V1 in people with autism. Is that right? No, a shorter intrinsic timescale in V1 in people with autism, and a longer intrinsic timescale is somewhere in prefrontal cortex. But then when you fit the model parameters, you actually found a stronger recurrent excitation in V1 in the autism group. Now, that's possible, but it would seem to go against the Chowdhury model. But maybe the reason this is because of some difference in the long range connectivity, or maybe there's some other explanation. But if you try to explain both the long range connectivity and the intrinsic timescales in the same model, I wonder, would you come to the same conclusion?
You point out really good, important points. For me, what is the already problematic is I always wonder what is the optimal structural connectivity that give you long range connectivity, and then depending on what kind of structural connectivity configuration you use as a backbone for your model, again the optimized parameters is also will be changed. So yeah, there are many factors that can affect it.
Also, I just want to just ask, maybe I don't think I have an answer, but just touch on Boris's question or point that, you know, should we be doing modeling, dynamical modeling, or should we just be doing spin test correlations? And I think it depends what your question is, right, or what your goal is. I think there's obviously a lot of value in spin test correlations. We've seen that from many people here. But, you know, I don't think it always gives you the answer, right, for all of the questions. So, for example, if we see a difference in the intrinsic timescale, you know, across the cortex, and we see that the intrinsic timescale correlates strongly with myelin or T1, T2 weighted signal, for example, right? Does that mean that that is because of changes in cortical myelin? Well, I think that if you collaborate with anatomists and physiologists and you try to think carefully about your modeling, well, maybe modeling can kind of try to provide answers there. So I think that there's a place for both, and I think it depends on what your goal is.
Thanks.
Okay. Yeah, I kind of agree with Sean's point about that. Yeah. But let us move on to more other people have a chance to ask questions. Mikhail did raise his hand, but I believe that there were other questions in the chat that came in earlier. So let's try not to neglect those in the chat as well. I think Daniel has a quick question for you, Sean. How much of your variance was explained by your first PCA, mostly for MPA and MDA?
Right, so the first PCA was completely dominant in the receptor per neuron data. It was almost 80% of the variance. And this is really not, I wasn't sure, I don't think I understand the AMPA and MDA bit, because this first PCA actually had contributions from all of the receptors. And this is why you see this extremely strong correlation with the total number of receptors per neuron across all the receptors. So this is essentially the way I see this, and maybe you can read the preprint if you're interested, is we also see this increase in the size of the dendrites of the pyramidal cells as you go up this gradient. That fits really well. So I think you see these pyramidal cells with big dendrites, and they have all types of receptors. And then it's, once you get into the second, third, fourth, fifth principal component, is where you start to get interesting patterns of different receptors expressed across cortical areas.
Okay.
Now,
Sophie's.
Yeah, so go ahead, Thomas. I think Sophie's had her hand up for a while, and then, but.
No, yeah, so no worries. You can also go for the chat. I just got a tired arm from raising my arm. No. Do you want to go first for the chat, or shall I ask the question?
No, Sophie can go first, followed by Kersia in the chat, and then Mikhail. How about that?
Okay, so yeah, first of all, thanks for your amazing talks and works, yeah, very, very cool. So I have a more broader question, because also Casey mentioned at some point breaking the structural model a little bit, and my question is also thinking of things in the brain that break the structural model. I was also thinking about the cerebellum, for example, in showing little cytoarchitectural variation, and then thinking further also about what these hierarchies or heterarchies mean. Do you think this is more of a mirror reflection of whatever happens outside of the cortex? So in the end, what does it, maybe this is a too general question, but what do you guys, does it represent? Is it an answer or just a mirror image of something that happens somewhere else, ultimately, beyond the cortex?
I can give it a go.
Yeah, Casey, this is a question for you.
So I think that I don't think these are mirror images by any means of what's happening in the subcortex. I think that would make them extremely redundant. So speaking, for example, of the default mode, I think that we see such expansion there because of the uniqueness that it offers relative to what's already happening, and that it can pick up on aspects of processing that aren't being used in subcortical areas. Obviously, Mac's really nice work on that really wonderful review about the cerebellum kind of picking up kind of the slack and doing a lot of automating for the cortex, for the rest of the brain. And then so you can have other types of combinations in these different networks. That's how I tend to think of them. They're all providing these complementary ways of recombining data, either based on different types of input data or the way that the data is recombined. But I don't think that they're just mirror images of subcortex.
And maybe also the same question for Seyok, do you think that what you see in ASD, is this a cortical issue or something that is beyond?
Well, because in my study, I should have actually included subcortical structures, but unfortunately I didn't. So I don't have a clear answer for that, but to me, it's always a mixture of between subcortical structures and the development of sensory networks at the initial developmental phase. Mm-hmm. And as the aging is ongoing, those effects from either from subcortical structures or sensory network is gradually accumulating to give effect to the high order areas. So in the end, I think it's probably both, but in my study, I didn't include the subcortical structure, so I cannot really answer as evidence-based.
And yeah, maybe also, then I'm finished. But like, I was also, like, you mentioned also the word hierarchy, and like, do you, in higher order regions, but can we really speak about higher order regions and not like, can we really see it in this more hierarchical way, or like how to, like, obviously it's a system, right, and it's organized in some way, but can we really prioritize like one system, like the default mode, as seating at the top of cognition? Because in some sense, Casey's argument is a little bit, because it integrates so many different cortical types, right? At least that's something that makes it special, I guess, or at least that's your point. But can we really say that one is, well, not better, but like, how can we then speak about the system if there's not a beginning and an end, right? It would be so nice to have it as a seat of cognition, sitting there with the crown being the default mode, or like in some way, but like, how do you, maybe also seen, because you mentioned the word hierarchy at some point, that's why I also had to think about this with relation to the subcortex and sensory regions. So
am I the one who should answer the question, or?
no, Sean, or you?
Oh, right, sorry.
Because you mentioned hierarchy, right. So that's why.
Yeah, sorry. Okay. I like the idea of maybe we can all get together and decide what the best network is. My vote is the default mode. I think Casey's agreeing, maybe Daniel. Yeah, so we define the hierarchy in by looking at patterns of retrograde track tracing data. So feed forward connections, so from V1 to V2 and V1 to V4 and so on, they tend to come from the superficial layers, and the feedback connections in the other way, they tend to come from the deep layers. And if you can put together a model then to predict the hierarchy of all of the areas that you have this retrograde tracing data for. So what I was correlating with, when I plotted the hierarchy, it was just this prediction based on the laminar pattern of connections. But what you actually see, I mean, for a start, I don't really think that the cortex is like, best described in a line. We have an updated version of a paper based focused on dopamine and working memory, where we show at least an updated version of a two-dimensional hierarchy, similar to stuff that Daniel's published and so on. So what I will say is that this method is based on the connectivity, but also if you look at the receptor data and so on, and most of the data I've looked at, it's pretty easy to separate early sensory hierarchies like V1, V2, V4, also in somatosensory cortex are pretty good. Once you get into like areas that we would consider part of the like higher cognitive networks, the hierarchies based on these like data-driven methods that I've used tend to be a lot flatter, and I'd have a lot less confidence in saying that, area 9/46D is above area 46D or so on. I think that's one of the nice things about Daniel's and colleagues' gradient paper is that it brings a bit of order into these higher cognitive networks. But a lot of the anatomical data that I've looked at in the monkey, we don't maybe have in the connectivity, for example, complete mapping of the higher cognitive network. So I don't want to make any kind of strong statements there. Definitely. Great question.
Yeah, thanks so much. Yeah, no, I think it's, I think a lot about hierarchy since I'm a group leader, and about how to like make organizations work, like having a flat hierarchy, because it's also, I guess, imagine like, not for my group leading skills, but like in the brain, tricky if you have no hierarchy, how to organize it then, right? Because there has to be something organizing, even if you don't have a hierarchy, I assume. But I'll let, I think, somebody else either in the chat or with the hand raised have a go. And thank you so much for the awesome work.
Let's go with Kersier's question, and then Mikhail will be probably the last question. Kersier asks, Seokju, I guess you can read yourself, I guess. He or she is asking how intrinsic neural time scale is calculated. Is it consistent with the work by Watanabe?
I just checked the paper to compare. I could have checked this first, but now I found this. It is shorter. What Watanabe found was shorter than what I found. Like my finding on intrinsic time scale is like from 2 to 4 somethings. To me, it's always remind me that it's related to the repetition time of fMRI, TL time, and also so it's not clearly represent the only neural activity, but it's sort of hemodynamically coupled signals, and then so this is bold dependent intrinsic time scale, and another one is how you calculate this intrinsic time scales also matter, because I used what I think John Murray used, like those decay length. But what Watanabe used is rather calculating the under the area curve of this autocorrelation function. So depending on what kind of measure you use, it also could be different. So yeah.
Okay, we've got a couple minutes left. Michal, would you like to jump in with your question?
Yeah, sure. Hear me?
There we go. Yes.
All right. So I've got a more methodological question about the diffusion embedding technique. So if you use the fusion embedding, you need to adjust the 2 parameters, the alpha parameter and the diffusion time parameter, or the other called scale parameter. So my first question is how to adjust them optimally, or can I just stick with the default values from the script that were probably used? And the second question is concerning the input matrix to the diffusion embedding technique, the affinity matrix. I noticed that instead of putting the connectivity matrix, there is an additional line of code where you calculated cosine similarity, like the product of this connectivity matrix. And I wonder why we cannot just plug the simple connectivity matrix instead. So 2 questions: alpha time and the matrix. Thank you.
Maybe I think more generally, if we may, and then of course I think there'll be plenty of time today to talk about different methods and comparison, but more generally, guys, any of the folks who spoke just now on the panel, what are your thoughts regarding the impact of different analytic techniques? Obviously, we're early on in this kind of field and application, and so there's a lot of space for methods comparison. Are there any insights you've had regarding the impact of these various parameters on your analyses, or how to best optimize for the selection?
So there are many important points that we should discuss. Just to answer his question first, I'm not sure I know that all of 2 parameters that you said, but one of them, if I know correctly, is about is related to how much you measure the Glover effect of your the data structure that you want to reduce the demand. For instance, if you use certain value for the parameter that I'm referring now, you rather consider more direct connectivity configuration. Whereas if you increase that parameter, you take care of much second or third order of connectivity, so that you wanna reduce the dimension, taking into account of this far fetching information in across the networks. So it's
probably the alpha parameter. It makes sense. It makes total sense.
Yeah, right. It depends on how much you care of this global topology, or you rather take care of the direct neighborhood connectivity configuration. I think.
So more like direct means closer, and more like distant.
Like
waiting.
first order neighboring, like a direct connectivity from functional, or if you want to take care of what is the neighborhood that I could visit through a multiple step, for instance, right? So if you want to take into account, a more global overarching framework, then you want to increase that parameter or decrease. I'm not sure which direction you should do, but it's related to that aspect, I think.
So I think that's more like to the diffusion time related, because diffusion times is making the scale of the data. How much the more diffusion time, the less clusters you get, something like this.
I think so.
Thank you for your insight. Yes.
I think when you use similar approaches to simulate functional connectivity from structural connectivity, as you increase diffusion time, your proxies become more and more polysynaptic. Perhaps that's like a good analogy for is that it's like not the direct path, but you really make the networks, you get something that looks closer to a default mode network actually than from diffusion MRI per se.
Okay.
But I think Sukjun has also done a cool study, Sukjun, where you evaluated the reproducibility of like all these different algorithms, and you also played around with using the time series versus the connectivity or the affinity matrices directly. That's published in NeuroImage from last year.
I would love to see this paper.
I'm gonna put in the chat, but there could be many ways to judge which algorithm or which combination of parameter would give you optimal result for the dimensionality reduction. But optimal is already like somehow operational definition depending on your goal. But my study, how I checked the optimality was 3 different conditions. One is reproducibility, reliability, and predictive validity. So like I made the study to try to give you the guidance, what kind of combination of parameter you should use to expect the gradient first. And then I checked these 3 conditions. And then, but I don't say only these 3 conditions are the condition that you have to think about, but as a basic set to develop robust biomarker using this gradient, these are, I believe, the 3 basic conditions. So.
Okay. Thank you very much. Do we still have a time to answer the question about this connectivity matrix versus? So I asked the question about why don't we do diffusion embedding on the connectivity matrix, but we need to do another process of this matrix to get the cosine affinity matrix.
So again, I mean, I'm sorry if I take all the time to answer this question. So in that paper that I referred, that I published last year, what I also evaluated was whether when I used the directly time series as input, or functional connectivity, or after calculating similarity network and then input, what is the optimal way to prepare this input is also can be evaluated by these 3 conditions. So indeed, in that paper, I checked these 3 conditions, whether using this time series give you a higher reproducibility, higher reliability, or higher prediction power. Then turns out that doesn't really matter, but it's more imperative condition is how much you threshold the connectivity metric, and without particular reason why you want to use nonlinear dimensional reduction. At least in my study, it turns out that the PCA is the actually simplest way, is giving you the higher reproducibility, reliability, and prediction power compared to other nonlinear techniques. So yeah, that's
okay. I'll definitely to check it out. Thank you very much for your insights.
Okay, we are running 5 minutes over. Lorenzo, I'm sorry, but we'll probably stop right here. Perhaps you can ask your question in the chat. And I'm gonna hand it over back to Daniel, and I'm gonna hand it over back to Casey.
Yes, I would take it. Thank you so much for running that session, Thomas and Daniel. That was really nice. And yeah, Lorenzo, please feel free to put your question in the chat. And if there's any leftover questions, many of us might still be around in the social at the end of the day as well. Or you can strike up conversation because you know us all now. Okay, so that means that we have very quick little interim, which is going to be gradients trivia. So if someone, I think Richard, could you drop the link for the form into the chat for me? Be my co-host. Thank you, Richard. Okay, is everyone ready? So all you have to do is click on that form and play along. It's going to be really fun and quite short. So we really want to see who knows their gradients, and we'll crown a champion by the end of the day. Okay, so just click the form. If there's any issues, let me know now. Otherwise, I'll power along. So the idea is that it's just a pretty typical trivia. I'm going to read out questions. There'll be 3 questions per section, 4 sections in total, I think. And you just write into the form your answer. Some are multiple choice and some are long answer. But because it's like a typical trivia thing, and I know you have your computer right there, but you know, I'll trust you that you're not going to Google anything. And that's also why the questions aren't on the sheet, so you can't just copy and paste anything and be sneaky. Okay, are we ready? Someone give me a thumbs up. Has anyone managed to access the form? Yes, thank you. Okay, first round. Round 1. How many years has the gradient workshop been running? Starting off easy. Okay. Oh, this one's great, because if you were paying any attention just then, you should know the answer number 2. What procedure is typically performed on a functional connectivity matrix to identify gradients? I'll take a range of answers here, but there's a couple of obvious ones from the first ones, right? Question 3. Fill in the gaps of the famed 1998 article by Marcel Meslem from blank to blank. Just fill in, what are those 2 words? From what to what? Okay, well done. You made it through round 1. Now, we are moving on to, if I can move my slide. Here we go. Is that the next round? Yes, that is. The next round is Know Your Neuroanatomist. So we have 6 different neuroanatomists here who are your multiple choice answers. They are Corbinian Brodmann, Franz Gall, Paul Flessig, Cecile Vott, Oscar Vott, and Konstantin von Economou. So for each of these questions, you have to choose which of those neuroanatomists fits the question. Okay? So question 4. Who had their habilitation thesis, cytoarchitectonic parcellation of the cerebral cortex in prosimians, rejected by the University of Berlin? The habilitation thesis was rejected, this famous neuroanatomist. Okay, question 5. Who undertook a lecture tour of Europe, espousing a theory's organology using pet monkeys and human skulls as props? So who espoused a theory of organology? Okay. Last one for the round. Which myeloarchitecture expert described the boundaries between areas as haarscharf Grenzen, sharp as a hair? I'm sorry for butchering that German. So who described boundaries between areas as sharp as a hair? Okay, next round, we have a picture round. All you have to do is name the first author and the year of publication for each of these famous gradients. So you get 1 point for naming the first author, and you get 1 point if you can also name the correct year. but I'll give you half a point if you can get it within 5 years. So that is questions 7, 8, and 9. I'll give you a few seconds. Okay, hopefully you have at least the ideas in your head, or take a screenshot quickly if you want to ponder it for a little bit more, or I can leave it up at the end as well. So let's move on to the final round. It might get a little bit harder, I warn you, but it's multiple choice. So which of these regions is identified as an origin in the dual origin theory? Is it the olfactory cortex, primary visual cortex, primary motor cortex? Which one is an origin in the dual origin theory?
We're gonna do it. Club 1.
Okay, and 11. Which mammal doesn't have a corpus callosum? Is it a whale, a platypus, or a sloth? Which one doesn't have a corpus callosum? Okay, final one. The natural coordinate system of the central nervous system, proposed by Rudolf Neuenhaus and Louis Puels, is based on what? Is it evolution, development, or connectivity?
Sorry, can you repeat the question, please?
Question 12? Sure.
Yes.
The natural coordinate system of the central nervous system, proposed by Rudolf Neuenhaus and Louis Puels, is based on what? Evolution, development, or connectivity? It's the natural coordinate system of the CNS.
I think it's evolution of the human race.
So just enter your answers on the Google form. We'll find out after the next session. I'll quickly mark all the papers. I'm not teaching right now. I'll quickly mark all the quizzes, and we'll see
Okay, sorry, babes.
who knows the gradients the best. Okay. So with that, that's our little grading quiz. I hope it made you think about some neuroanatomy. And it wasn't
Thank
too
you.
hard.
Can I, you got WhatsApp?
Okay, so what we're going to do is take a 7-minute break roughly and come back and return here at 10 a.m. And then we'll get started with session 2, which will be chaired by Boris and Beth. Okay, thanks everyone.
Hi, Beth.
Hello, Boris. How are you?
Yeah, nice to see you. Yeah, cool. Thanks
Great.
for joining.
Oh, it's my pleasure. What a wonderful afternoon it's been so far. Really, really great. Yeah. How are things with you? Are you all right?
Yeah, it's good. Yeah, home office still, but you know, it is what it is.
Yeah. Are your kids still at school? Are they able to go to school?
Yeah, they kept schools open most of the time. Just last year, there was like a little bit of an interruption, but yeah.
Oh, what a relief. Yeah.
Yeah, are your kids back already?
They're at school, yeah, but they're getting older now, so they look after themselves mostly, so that's good.
Okay.
But yeah, they are at school. I'm just thinking they're gonna sort of show up. So I'm hoping they'll keep the noise down. And stick a sign on the door saying "Be quiet." But they're coming home from school in a few minutes.
Yeah, I have that too. Sometimes at 4 o'clock, the noise level in the house just changes significantly. So
yeah, fantastic. So this is the best organized meeting I think I've ever attended. It's extraordinary. It's so smooth.
Well, Casey, Debbie, and the events team, they have us in good, we're in good hands, basically. So.
Amazing. Can you remind me, Boris, so are the next ones pre-recorded or are they giving live talks?
I think it's like a mix of both. I'm not 100% sure who pre-recorded and who will give live.
Okay, we'll find out.
We'll find out. I think we'll always introduce like one of the speakers, and then the talk will either run or they will give it live. And after that, there's like room for one question, and at the end we'll have like a general discussion question period.
Deborah's saying people are coming back. It's funny when people's cameras and microphones are off because you can't tell.
Yeah.
No or not. We are live. Hi, Casey.
Casey, did you put a fan on? Did you get a fan?
Yeah.
You have like this wind. You have like this wind effect.
This whole wind machine going. It is so hot here. But I just want to say hi and fill this black void. And if any of the speakers are around, you can turn your cameras on and join the crowd as well. But otherwise, feel free to start whenever you like, Beth and Boris.
Okay. Yeah, Beth, do you want to take it away?
Yep. Shall I start now? So it's with great pleasure. Do you want to, actually, Casey, do you want to put the slide up with the talks, the next?
I don't have it, but Sasha might have it in control. There we go. Oh, well, the next talk is ready to go, I guess.
Okay, well, that's fine. So thank you so much to the organisers for this really fantastic afternoon where I am. I'm really, really enjoying myself, and it's wonderful to hear these amazing talks. So in the next session that's about to start, the talks are going to be focused on dynamics and cognition. So I'm Beth Jeffries, and I'm co-chairing with Boris this session. And there are 4 talks coming up. They all look at different aspects of network dynamics, things like harmonics, oscillations, dynamic fingerprints. And we've also got a talk about multiple gradients near the top of the hierarchy in the anterior temporal lobe. So I'll just hand over to Boris to introduce the first talk.
Yeah, thanks, Beth, for co-chairing this with me, and thanks, Casey and Debbie, for doing this terrific organization of the workshop. So it's really great. Thanks to all the speakers also for joining in. I hope this is going to be an interesting session. The first speaker is Katharina Glomp. She did her BSc in biology and an MSc in computational neuroscience in Berlin at the Humboldt University and at the BCCN, the Bernstein Center for Computational Neuroscience. She did her PhD with Gustavo Deco in Barcelona and is now a postdoc with Patrick Hagman, where she's working on establishing techniques to link structural and functional connectivity, leveraging MRI as well as EEG. And today we hear about network harmonics and the structure-function relationship in fast network dynamics. Thank you so much, Katharina. Looking forward to your talk.
Hi, yeah, thank you to the organizers for giving me the opportunity to present our work here. And today I'm going to show you a little bit of our recent studies on network harmonics and the structure-function relationship, specifically for fast network dynamics. First of all, I've moved away from using the word network harmonics and I try to stick to harmonic modes, but yeah, both of them will kind of come up, and just so you know, it's the same thing. I work mostly with EEG data these days in a multimodal setting, so we project the EEG data recorded on the scalp into the source space, so then we have brain activity in the actual brain space, so that we can also combine this with other modalities from MRI, specifically diffusion MRIs, white matter tracks, and our usual white matter connectivity graph. I emphasize this distinction between the underlying graph and the signal on the graph that evolves on the graph, because I will be talking about gradients, aka harmonic modes, in the context of graph signal processing and what you can do with those in graph signal processing. So again, there's a clear distinction. The signal, the EEG signal, evolves on the graph, which is obtained from MRI. And why is this important? It's because harmonic modes are Fourier basis functions in the same sense as sine and cosine waves are Fourier basis functions of the circle. So usually if you have a one-dimensional signal like a time series, but you can also think of a wave pattern in space, like on a string, a vibrating string, something like this, then you can decompose your signal into usual sine and cosine waves of different frequencies. And now your domain on which your signal involves is not a circle, not a regular domain, but it's a graph. It's structural connectivity graph in this case. And if you obtain the Fourier basis functions of this graph, you get harmonic modes, aka network harmonics. And what are harmonic modes or Fourier basis functions? Well, so they are building blocks obtained from the white matter graph. And these building blocks can be used to reconstruct the functional signal on the brain surface. So in this case, EEG signals. And that's what we did in this paper from last year. So what did we do there? We used harmonic modes for the tracking of fast network dynamics. What we used was EEG data from 19 subjects. Again, source projected to the gray matter, so this is actual brain activity. And these were healthy participants who were presented with images of faces, and they just had to detect this face and say if they saw a face or not. So we start with our ROI signals, where on the y-axis you have brain regions, and on the x-axis you have time, of course. At 0, the stimulus, so the faces are presented, and then you have your usual brain activity all over the brain in your different brain regions. And what we can do with harmonic modes is you can transform your activity, your ROI signals, into this harmonic modes or graph space, and then interpret this as a sort of network activity. So if you interpret your harmonic modes as networks, then this is how you can get at the network activity. It's just a matrix multiplication where so the SE with the hat here is your network activity, and all you do is you project your EEG activity, so your functional signal, onto your harmonic modes, which are obtained from structural connectivity. So here you have this multimodal aspect where you combine these two modalities. So this is the transformation into the graph spectral domain. And just to make it clear again, this is different from the usual Fourier transform, where we have a transformation from time to frequency domain. I am talking about a transformation from node to graph space. So on your x-axis, you still have time, you still have the same stimulus presentation and processing as before, but on your y-axis, you no longer have brain regions, but you have harmonic modes, or yeah, here it says network harmonics. So you have your networks. And yet, as a little teaser, of course, you can do both at the same time. So what we did with this, we analyzed our visual evoked potential from the phase detection task. Here, the blue line is just the average over all trials, over all subjects, and the gray lines are each single subject. And what we did was we transformed it from node to graph space, as I just explained. And what happens is you get this colorful thing. So as with the usual Fourier transform, because we have 68 brain regions, we also have 68 harmonic modes. They are also, like in a usual Fourier transform, ordered by frequency, in this case spatial or graph frequency, so not temporal frequency. And also another thing that should be pretty familiar is that the signal power is concentrated in the low frequencies. What we did next to make this whole thing more interpretable, because all we did now was to transform from one 68-dimensional space into another 68-dimensional space, is we applied a statistical test to get rid of the harmonic modes that did not change significantly from before the stimulus to after the stimulus presentation. And we ended up with a small number of these harmonic modes that were significantly activated by the stimulus and by the stimulus processing in a small number of time windows in which something like this occurred. So what is the nice thing about this? Again, you can see that this network activity reflects the visual evoked potential. And we have these different time windows in which we have significant changes compared to baseline. And when you now reconstruct your activity again, so this is just the inverse Fourier transform now. So you reconstruct your brain activity from these network activities that you obtain from the statistical test. So you go back from your graph space to your node space, and you can project this again onto the brain surface. Then you clearly see that using this approach, you get a good characterization of what happens when people look at faces. So at first, there's the activation of the visual system, in particular the fusiform face area, so where people process faces and recognize faces. Then at a slightly later time point, so in this period, we think that decision-making takes place. So this is frontal regions and default mode network regions that are involved in these networks. And later on, people start preparing their response. So they press a button. So here we see somatomotor regions. Okay, now I would like to come to a slightly different point about this whole business. So as I said before, signal power is concentrated in low frequencies. And why this is interesting is that in low frequencies, the patterns, so these building blocks that I showed before, these ones, they're smooth, as you can see. For example, the first one includes the entire brain. The second and third one just kind of divide the brain into two big chunks. So we interpret this as being more related to integration. While if you look at a higher order building block or harmonic mode, so the high frequencies, the high graph frequencies, they are less smooth. For example, here I'm just showing the last one, the 68th one, and you can see that clearly most of the brain is not even involved in this component, and you just have these two brain regions which are doing completely opposite things. So they're segregated from each other, so we interpret this as being related to segregation, segregated specialized processing. So now I want to talk about no longer this paper, but a new paper which is under review right now and which was a project led by Joan, this young man here. We hope that it's going to be published soon. So here I want to still talk about the same visual evoked potential. I'm going to compress it a bit in this slightly ugly way, just to show you a figure from his paper. So here we divided the harmonic mode spectrum into the high and the low frequencies, as I just explained, so related to integration and segregation. I'm just going to show a normalized version of that because then it's easier to see. So these two pictures are the same, except the curves are normalized to always have the same power. So you can clearly see these different periods of time where the high or the low frequencies are dominant. And again, this is related to the different processing stages of the visual evoked potential, which I also mentioned earlier. Namely, in the beginning, the initial visual stimulus processing, here you can see that the low frequency components, so the low spatial frequency components dominate. And the same happens later in the decision-making phase. And in the middle, you have the actual face detection, which is a very specialized task. So this is where the famous N170, for those who have heard of this, occurs. So people recognize the face. And this is specialized, so we have the high frequency components contributing more. And we interpret this as being highly integrative processes versus a highly segregated process, which involves the activation of just a small set of brain regions. So to summarize this, harmonic modes are a useful basis for representing source-projected EEG data. So maybe you've noticed that we have never computed functional connectivity at any point. So it's pretty simple. We can get around all these different connectivity measures, which are very problematic in EEG. The whole thing is multimodal. And by the way, it's also model-driven because it assumes that the EEG signal evolves via diffusion through this underlying graph structure. And it's compact, interpretable, and statistically powerful because we're directly analyzing networks, so sets of regions and not single brain regions. And as I showed you in the end, these harmonic modes also reflect the hierarchical properties of the connectome. So there's this ordering from low to high frequencies, and this is how we can use them to also relate them to integration and segregation without ever computing functional connectivity. Okay, this is all the time I have today. I would like to thank you for listening, and I am now at the University of Tübingen, but all this work was done in my previous position at the CHUV and the UNIL in Lausanne with the Synergia Consortium on brain communication pathways in the Connectomics lab of Patrick Hagmann. And yeah, I also want to say thanks, Hélène, for getting me into this whole harmonic modes and connectome harmonics business. And yeah, if you feel like discussing or have any more ideas, you can always send me an email at this address down here. Thank you.
Thank you very much, Katharina. That was fantastic. So we have some time for one question or two. I may read like one of the questions from the chat. Just a clarification of which source imaging technique you use to estimate the source signals.
Yes. Can you hear me?
Yep.
Because I can't see anything. My Zoom has been frozen for hours. I'm actually watching on Vimeo. But okay, so we used a bit of an obscure software toolbox called CarTool, and in this one, yeah, you can use the different styles of source reconstruction like Laura or Loretta or whatever, but we use Laura in this case. Does that answer the question? We've tried with Loretta, and the results are robust against this.
Okay. Great. I have like maybe one quick question because at one point you also showed this interaction between the order of the harmonics and the smoothness or the distributed nature of the components that you get.
Mm-hmm.
Is that something that one would naively expect from like any kind of decomposition technique to get such a result, or would you think this is explainable by, you know, temporal autocorrelations, spatial autocorrelations of these signals?
I haven't thought of it in terms of autocorrelation, but I know that in another paper we compared these components that you get. So these components are obtained from the graph Laplacian, eigenvalue decomposition of the graph Laplacian. So yeah, you explicitly use kind of the gradients between the brain regions. And we compared this to like PCA and eigenvalue decomposition of the functional connectivity matrix, and you do not get this ordering. So this ordering by smoothness is, by definition, it's part of this graph Laplacian approach. Yeah, you get it from the eigenvalue decomposition, and it's really kind of the sum over the differences between the neighboring brain regions. So the higher you go, the higher order your gradient / harmonic mode / network harmonic is, the bigger the differences are that you allow between connected brain regions. And this is where you get more or less smooth, more or less global or local patterns. So yeah, it's specific to this kind of gradient approach, let's say.
Okay. Yeah, cool. Thank you. I think we can, we'll go to the next speaker now.
Yes.
And then there will be
Thank
some
you.
time for question and answers at the end of the session.
Okay, so I'll introduce the next speaker, Keivan Majori. So Keivan is a postdoc at the Max Planck Institute for Empirical Aesthetics in Frankfurt, and has a background working in EEG source localization and connectivity, and did a postdoc at the Institute for Biomagnetism and Biosignal Analysis. So we're delighted to listen to your talk now. Thanks very much.
Hello everyone, thank you for joining into my talk, and a big thank you to organizers of this workshop for inviting me to be one of the speakers. My name is Keivan Majori. I'm a postdoc researcher at Max Planck Institute for Empirical Aesthetics. I will present results of our recent paper, The Frequency Gradients of Human Resting State Brain Oscillations Follows Cortical Hierarchies. So it has been shown that many structural and functional features of the cortex change gradually in an orderly manner across the cortex and form gradients. The most prominent gradients are in the posterior anterior axis. On the other hand, frequency of brain oscillations has been related to many functional features of brain activity and has been shown to be characteristic for each brain area, similar to many structural features. But it is rather unknown how the frequency of brain oscillations change in the cortex across brain areas. So we hypothesize that spatial gradients of cortical features are associated with a similar gradient in a prominent feature of brain activity, the peak frequency of brain oscillations. And to test this hypothesis, we used public MEG data from Mouse Project. Data contains 5-minute resting state MEG data from 204 participants, together with MR images. Analysis details. We pre-processed the MEG data. From MR images, cortex surfaces were extracted to solve the forward model. Then we reconstructed the cortex level time series from MEG sensor time series. Cortex surface was parceled into 382 regions of interest, and then time series for each of those regions of interest were extracted. Roy time series were reshaped to 2-second epochs, and power spectrum was obtained. Then we applied dimensionality reduction over all power spectra to obtain one power spectrum per row and per participant. Then we identified peaks from that power spectrum. Here you see an example for power spectra of a row. So we 1OF component of power spectra was estimated and subtracted from the original spectrum. All peaks on the 1OF corrected spectrum were identified, and the peak with the largest amplitude was selected as the peak frequency. So that's our definition of peak frequency here. First, we were interested to see how peak frequency is distributed in the posterior anterior axis. Here you see distribution of peak frequency as a function of the Roy's location along the y-axis of the coordinate system, or posterior to anterior direction. And each point represents the mean of peak frequency across participants for a certain row. So as you see, a clear gradual decrease of peak frequency from posterior to anterior is evident and supported by a significant correlation. So that was a pattern for the mean of peak frequency. Next, we wanted to analyze this pattern at the individual level. So for each individual, we computed the correlation between peak frequency values and y coordinates. So each point here shows that correlation value, and found a significant posterior to anterior gradient of peak frequency for 84% of participants. So this plot nicely shows consistency of peak frequency gradient, but also it shows variability of gradient between participants. To account for this variability and also assess the changes of peak frequency across the entire cortex, not only the y-axis, we applied a comprehensive statistical model using mixed effect modeling. So we model peak frequency as a function of Roy's location, or a detailed version of the equation where peak frequency is the response variable and the coordinates of the Roy's location x, y, z plus their two-way interactions are the independent variables. Here x is defined from left to right, y is the posterior anterior, and z is the inferior superior direction. And these S terms here are to account for variability in the slope of gradient across participants, or our random effects. And the term here is to account for the variability of peak frequency variability of offset of peak frequency between participants. Again, random effect. So this model found a significant gradient of peak frequency along the y direction and z direction and their interactions. Here you see, and here you see the fixed effects of the model, or the model's prediction of peak frequency gradients. So these results support the conclusion that peak frequency of brain oscillations decrease systematically in posterior to anterior direction. And as we saw earlier, neural power spectrum consists of spectral peaks on an 1OF component. So variations in properties of that 1OF component can bias our estimates of peak frequency. So here we first examine the spatial distribution of 1OF offset and slope across the cortex, and found significant scores for y-axis, z-axis, and xy and yz, and along the y-axis, xy-axis, and yz. Then we tested to what extent the peak frequency gradient is independent of spatial changes of 1OF slope and offset. Again, we used linear mixed effect model and regressed out the linear combination of 1OF offset and 1OF slope to obtain residuals of peak frequency, or 1OF corrected peak frequency. Then again, we modeled 1OF corrected peak frequency as a function of Roy's location. As you see, the gradient, the posterior anterior gradient, holds after removing the impact of 1OF component. So the posterior anterior peak frequency gradient is largely independent of observed gradient for 1OF components. Then we were interested to see if this peak frequency gradient follows cortical hierarchies as well or not. It has been shown that cortical thickness mirrors hierarchical organization of cortex and therefore can be used as a proxy for quantification of hierarchical level of brain areas. So we computed cortical thickness for all rows and all participants. Here you see the mean of cortical thickness that shows a significant gradient along the posterior anterior axis. Similar to previous analysis, again we used linear mixed effect model and found cortical thickness as a function of location and found the significant changes in the posterior anterior and inferior superior axis. Next, we applied linear mixed effect model between peak frequency and cortical thickness and found a significant negative correlation between them. But one may ask if this relationship is driven by spatial gradients of peak frequency and cortical thickness, or the question could be asked in a different way, that how would be the relationship between peak frequency and cortical thickness if we remove the effect of location from both peak frequency and cortical thickness. To test this, we applied linear mixed effect model between peak frequency and location to obtain peak frequency residuals or location corrected peak frequencies. The same analysis was applied to cortical thickness to obtain location corrected cortical thickness. Then we again found a significant relationship between corrected peak frequency and cortical thickness. And here you see the relationship between mean peak frequency and mean cortical thickness before correction and after correction. So peak frequency and cortical thickness are more directly related to each other than can be explained by their individual dependency on location. This result suggests that peak frequency is related to structural features that likely represent cortical hierarchies. So this study is the first comprehensive demonstration of frequency gradients across the human cortex using a large number of participants. We found that dominant peak frequency in a brain area decreases significantly, gradually, and robustly in the posterior anterior axis. Our results are consistent with a recent invasive study showing systematic decrease of peak frequency from posterior to anterior brain areas in ECOG recordings of epilepsy patients. And we showed for the first time that frequency gradients follow cortical thickness more closely than can be explained purely by spatial location. So I want to end by acknowledging the supervisor and the collaborators of this study. Thank you so much for listening.
Thank you for that really wonderful talk. There are a couple of questions on the chat if we've got time. So the first one from Suzanne, thanks for the interesting talk. Would you expect a similar peak frequency gradient during task performance? Are you there, Kayvan? You might be muted.
Hi, do you hear me? Yeah. Do you hear me? So I couldn't switch on my camera actually. Yeah. Yeah. Hi, do you hear me now? Yeah.
Yes, perfect.
Yeah, so first I should apologize because this version of video, in this version, the mouse pointer was not saved. So it was difficult to follow. Then I had sent a newer version, but yeah, for some reason, the old one was playing. Yeah.
So would you expect a similar peak frequency gradient during task performance? Have you looked at different cognitive contexts for this gradient?
Pardon?
I'm just reading the question from the chat from Suzanne, and she's asking if you'd expect a similar peak frequency gradient during task performance.
Yeah, that's actually an interesting question. We wanted to test it on other data sets other than resting the state to see how the peak frequency gradients are. And that's something that we want to do. An interesting question, yeah. And also in aging, how it changes over aging. That's another interesting question. Yeah.
Absolutely. Okay, so there's a few more questions in the chat. We probably haven't got time to cover all of them, but we could look at the next question from Makoto. So although it's interesting to detect a peak frequency from broadband signals, which are going from alpha frequency in your data, what would happen if you changed the frequency range, if you looked at delta, theta, beta, or gamma? Would the source map be the same or different? Especially alpha power would seem to change more dynamically in the visual cortex, consistent with your findings.
Yes. So actually, we analyzed gamma band, delta band, also larger, broader band. But the problem is that our estimates of peak frequency is based on 1OF subtraction. And 1OF fails in some cases. For instance, when you go above 50 hertz or below 3 hertz, then the 1OF is not linear in log scale. So that was one reason 1OF fitting fails in some cases. So then peaks are not detected precisely. That was one thing. Another thing that in gamma band especially, peaks are noisier and challenging. It's really challenging to pick one peak over all peaks. And another thing maybe to mention is that we wanted to find peaks that are consistent or more consistent across participants. So we didn't find gamma or delta peaks more consistent across participants. And it was, again, challenging to find something systematic across participants or across brain areas at gamma or delta bands.
Okay, that's great. Thank you very much. Oh, and there's a quick clarification. It's MEG data, wasn't it, that you were analyzing?
Yeah, it was. It was MEG.
Yeah, fantastic. I'll pass on to Boris now for the next talk.
Yeah, thanks. Thanks, Beth, and thanks for the great talk. So it's my pleasure to introduce the next speaker. That's Golia Shafiai. She's a PhD student at McGill, supervised by Bratislav Mizek at the Network Neuroscience Lab, and she holds an MSc and a BSc in physics. A true scientist, that's great. And so today we'll hear about the interactions between local dynamics and global brain dynamics and how they translate to inter-individual differences in behavior. The title of the talk is Topographic Gradients of Intrinsic Dynamics Across Neocortex. Thanks for being here, Golia.
Hi, everyone. My name is Golia. I'm a PhD candidate working with Dr. Bratislav Mizek at the Network Neuroscience Lab at MNI. Today, I'm going to talk about topographic gradients of intrinsic dynamics across neocortex. So when we study the brain as a complex network, we usually start with some parcellated neuroimaging data, and we use a graphic construction of the brain to divide the brain into nodes and edges. These nodes represent brain regions, whereas the edges represent the interactions between pairs of these regions. For example, the edges can measure white matter anatomical connections connecting pairs of brain areas to one another, as shown in the structural connectivity networks. But they can also represent statistical dependencies between functional time courses of these brain regions measured using functional imaging such as fMRI, and as shown in functional connectivity networks. Usually in this graph representation of the cortex, we assume that nodes are identical across the cortex, and usually we focus on the edges and what they represent. However, we know that in reality, there is this regional heterogeneity observed across the cortex. So for example, if you take nodes A and B and look at some of their properties, such as their morphometric features, their cytoarchitectonic properties, transcription or temporal features, we see that all these properties vary across the cortex. And interestingly, what we usually observe is that most of these properties follow a hierarchical organization, going from unimodal areas to higher order transmodal regions. This hierarchical organization was first proposed by Mesulem in 1998, where they proposed a hierarchy of processing from distinct unimodal areas to higher order integrative transmodal regions using track tracing studies in the macaque monkey. Later on in 2016, Daniel Margulies and his colleagues used a technique called diffusion map embedding on resting state functional MRI data in human brain. And they also identified this hierarchy in functional connectivity profile of brain regions, sort of separating unimodal areas from higher order processing areas according to their connectivity profiles. This is not only the case for connectivity and large scale connectivity, but also in other measures such as myelination, semantic processing properties, and also temporal properties. So today I'm going to mainly focus on temporal properties and on regional heterogeneity of cortical dynamics in the human brain. So we know that regional biological and structural properties such as gene expression and myelination interact with local circuit properties. And all these together are reflected in local brain activity as we can measure in time series in a regional time series signature, which is what we basically focused on in the study that I'm presenting today. So we wanted to focus on time series properties and sort of get to its relationship to other measures in the brain, such as biological and structural properties. So for this purpose, we, instead of selecting one or a few features, we wanted to extract a large number of properties, or take a data-driven approach. And we used a Human Connection Project dataset, and we took a resting state functional MRI data of a subset of healthy young adults that had no familial relationship with one another. And in order to extract a large number of time series properties, we used HCTSA toolbox, or highly comparative time series analysis toolbox, developed by Ben Fulcher and his colleague, which is a toolbox that one can use to sort of perform this massive temporal feature extraction of time series, and in our case, regional fMRI time series. So we have this sort of regional time series for the whole brain, and what we do is we apply HCTSA to it, and we extract over 7,000 time series features for each time series. So we end up with this region by feature matrix where each feature is a time series feature. Some examples of these features are measures that have to do with the distribution of time series data, measures that have to do with autocorrelation of the signal, and other properties. So we performed two sorts of main analysis here. One was to sort of investigate and to look at the pairwise similarity of time series properties across the cortex. So looking at temporal profile similarity. And the other one was to sort of see how correlated features, groups of correlated features, are distributed across the cortex. So starting with interregional temporal profile similarity, we sort of quantified how similar temporal properties or time series properties of pairs of brain regions are using this temporal profile similarity matrix, and we compared this similarity network to other aspects of large-scale network organization of the brain, for example, to Euclidean distance. And what we observed here was an exponential negative relationship between Euclidean distance and temporal profile similarity, meaning that the closer two regions are to one another, the more similar their time series properties are to one another. We also compared temporal profile similarity to structural connectivity networks, where we observed that regions that are connected to each other with an underlying anatomical connections have higher temporal profile similarity compared to regions that are not connected to each other. And this sort of difference was significant when we used null models, rewired null models, where we sort of randomize the network connections based on the edge length and the distribution of the structural network. We also compared temporal profile similarity to functional connectivity data, where we observed that time series properties of regions that belong to the same functional system, so within network temporal profile similarity, is higher than between network temporal similarity. So it's higher from time series properties of regions that belong to different networks. And again, this was significant when we compared this empirical sort of difference to a null distribution of differences obtained using spatial autocorrelation preserving null models or spin tests. We also directly compared temporal profile similarity to functional connectivity using a whole brain edgewise comparison, where we observed a positive trend between the two. So the higher the functional connectivity, the higher the temporal profile similarity. And this positive trend sort of survived this distance correction when we removed the distance effect from both matrices. So with this, we sort of observed that interregional temporal profile similarity reflects network geometry and topology. The next thing we asked was so we wanted to investigate the topographic organization of time series features across the cortex, and we sort of wanted to use principal components analysis or PCA analysis for this. And we wanted to identify groups of correlated features that explain maximal variance across the cortex. So applying PCA to our data, to the time series features, we observed, so we looked at the two first components, capturing more than 70% of the variance in the data, and we looked at the spatial distribution of these correlated sort of time series features across the cortex. And we saw that the pattern captured by PC1 sort of spans this posterior mediotemporal axis, whereas the pattern captured by PC2 represents, or it's like it displays this familiar sort of hierarchical organization we observed in the initial slides, going from lower order unimodal areas to higher order transmodal areas. We also wanted to know which features contribute the most to these spatial patterns. And for that, we looked at PC loading, so looking at the correlations between each feature, each time series feature, and the patterns captured by principal component analysis. And we ranked these loadings, and we sort of performed these statistical significant analysis using spatial autocorrelation preserving nulls. And what we realized was that features that contribute the most to PC1 have to do with autocorrelation of the signal, whereas features that contribute the most to PC2 have to do with how time series data are distributed, so distribution of time series data points. Just to give some examples of what these features are, we can look at high loading features of, for example, PC1. And looking at lag1 autocorrelation and nonlinear autocorrelation, we see that these maps sort of closely follow the pattern captured by PC1. And if we take a region that has a high PC1 brain score, as shown in on the brain here with this purple point, and we look at its signal, its time series, we see that it has a high autocorrelation, whereas if we take another region that has a low PC1 brain score and look at its signal as shown with this pink point and this pink signal, we see that it has a very low sort of autocorrelation. For PC2, if we look at some high-loading features such as keratosis and distributional entropy, we see that these patterns, these two example patterns, sort of negatively contribute to the pattern captured by PC2. And if we can focus on keratosis, for example, so keratosis is a measure of the spread of a distribution, or how the data is distributed around the mean, or a tailoredness of the distribution. So if we look at keratosis, and if we take a region from that has a low PC2 brain score, so it is a lower order sort of sensory area, as shown in this pink point here, for example. And if we look at this keratosis, we see that it has a pretty wide distribution, so it has a very high sort of keratosis. Whereas if we take another region that has a high PC2 brain score, so it is a higher order area, such as the one shown with this pink point here, we see that it has tighter distribution. So PC2, so for example, with PC2, looking at keratosis, we see that regions that are lower in the hierarchy sort of have this wider dynamic range, whereas areas that are higher in the hierarchy have this tighter dynamic range. We also, having seen this sort of hierarchical organization of intrinsic dynamics captured by PC1 and PC2, we wanted to compare these patterns to other measures of hierarchical organization across the human brain, such as principal component of gene expression, functional hierarchy, that comes from functional connectivity data, T1 weighted over T2 weighted ratio, which is a proxy measure for myelination, and also cortical thickness. So comparing these maps, we observed that there is a significant positive relationship between PC1 pattern and principal component of gene expression. However, a PC1 brain pattern wasn't correlated with other measures. But when we looked at PC2 as it sort of represents this like unimodal transmodal hierarchy, we see that it's significantly correlated with all these other measures. So PC1 mostly reflects the principal component of gene expression, whereas PC2 sort of recapitulates this unimodal transmodal hierarchy. So spatial variation in temporal dynamics sort of reflects this functional and microstructural hierarchical organization in the brain. We also compared the patterns captured by PCA with functional activation patterns. So we obtained these meta-analytical functional activation maps for 123 terms using a NeuroSynth database. And we compared our PC patterns to NeuroSynth maps. And we looked at, we sort of tried to identify the cognitive terms that their functional activation maps closely follows our PC patterns. What we observed here was that PC1 intrinsic dynamics sort of, so it mainly defines this cognitive affective axis separating measures such as attention from stress and fear, whereas PC2 pattern sort of defines this sensory cognitive axis separating measures such as perception and multisensory processing from sort of higher sort of cognitive measures such as cognitive control and memory retrieval. So spatial gradients of intrinsic dynamics support these distinct functional activations. So in summary, in this study, we saw that a dynamic fingerprint of regional brain activity is related to the macro scale network embedding. Pairwise similarity of local dynamics reflect brain network topology and geometry. Spatial variation in intrinsic dynamics reflects functional and microstructural hierarchical organizations and support distinct functional activation patterns. And local brain dynamics are hierarchically organized across the cortex and nicely recapitulating the unimodal and transmodal hierarchy. So with this, I want to thank you all for listening, and thank everyone in the lab and our collaborators and the funding agencies. Thank you.
Thank you very much, Gulia. Terrific data and terrific talk. So we have some time for like 1 or 2 questions. I can read the first one. Or Beth, ask it directly.
Hi, that was such a beautiful talk. Thank you very much. I was just wondering about how you interpret the difference in ketosis and whether the sort of wider distribution at the top end of the second gradient might be to do with the opportunity of integration of different responses happening at different temporal scales, for example.
Yes, so can you hear me or?
Yes.
Yeah, okay, awesome. Yes, so first of all, thank you very much for having me as a speaker in the workshop. It's been very nice. I've been following since the morning. And to answer your question, so yes, so what we think is that, so we are kind of like terming this as dynamic range. So sort of having a more variable values for in the time series. So and what we kind of hypothesize, like what we think is happening here, is that because lower order sensory areas are kind of in contact with outside world, so they kind of have like these different types of signals coming in, and they have to sort of like process all these different signals coming from outside. So that's why they have like this more like sort of dynamic range. So they could be like more entropic, having to process this like higher amount of information. Maybe I could say that that's what we think is happening compared to higher order areas that have like a tighter dynamic range. I'm not sure if I was able to answer your question, but
that's perfect. Thank you.
Yeah. Golia, that's like maybe just a naive question, because we also had like some electrophysiology or MEC studies before. What kind of acquisitions do you need with fMRI to address dynamics well, or what would be like a good technical development for fMRI to go forward, like even to go sub 100 milliseconds, or is like HCP type quality, is that enough?
Right, that's a great question. So the nice thing about HCP is that it has a relatively high sampling rate, like a good temporal resolution compared to most of fMRI data sets. But as you said, it's like neuronal activity is happening in a much finer temporal resolution. So I don't really have like a solid answer right now. But what we are currently working on is we are using MEG data set from HCP and sort of trying to look at temporal measures from resting safe MEG signal and kind of compare it to what we get with fMRI because although the modalities are very different and the time resolution is very different between the two, so we would expect to probably get different time series properties, but as they are coming from the same underlying kind of system, so the data is produced by the same underlying system, we expect to have some correspondence and some overlap between the features. Mm-hmm. So I would say in order to probably get to a neuronal activity, higher resolution, so higher temporal resolution, so faster sampling rate would probably be better because it would be closer to what is really going on with the neuronal activity. But to answer that question, I think it's like what we are currently working on is to looking at MEG signal and kind of trying to see if the features overlap with one another, or like which features have a higher overlap between the two modalities.
Mm-hmm. Yeah, that's maybe like also like maybe a broader question for the discussion at the end, but you know, there's like a little bit of a disconnect between like these sensory fugal patterns that you often get with fMRI and then this more anterior posterior gradients that we saw, for example, in K1's work. And I know like the modalities are different and the measures that are derived are different as well, but it would just be interesting to see what's kind of the, you know, mesolam sensory fugal versus anterior posterior hierarchy. Which one is the dominating gradient, so to speak?
Exactly. Yeah.
Interesting. So there's maybe like one more question from Sean. Sean, do you want to ask a question directly? You might be able to.
Sure. Beautiful work. I just was interested in a little clarification on how you interpret the first principle component, because it was related to the temporal autocorrelation, but I was wondering, do larger values in the signal, so the areas that were shown in red, do they have a slower intrinsic timescale in the autocorrelation function?
So thank you for that question. So what we observed was that most of the features that were contributing to PC1, they had to do with kind of lower order autocorrelation, so having a smaller time lag. So as we, for example, we also like looked directly at autocorrelation increasing the time lag, and we saw that autocorrelation is like kind of changing and going to, it kind of shows this unimodal high transmodal separation when we reach about 50 seconds, not 50 seconds, like 15 time points time lag in if we measure the autocorrelation with 15 seconds, with 15 time point time lag. So we get to this like unimodal transmodal hierarchy. So it's changing depending on the lag of autocorrelation. What I could say is looking at intrinsic time scales that people measure in, especially with make data, we also see this like posterior kind of maybe like medial temporal separation, but they also are able to capture unimodal transmodal hierarchy in the same brain map. So they have this separation between unimodal transmodal, but they also, like if you look at the maps, it also kind of goes from this posterior to a more like medial temporal area. So it's like both PC1 and PC2 maps, but in one map, I would say. So it's like both gradients are captured. So definitely they are related, I would say, because intrinsic time scale can be measured using autocorrelation measured with different lags. So they are definitely related, but I would say what we got with our PC1 was lower order autocorrelation measures that were highly contributing to that, to like the pattern we got with PC1, but they are definitely related.
Yeah, great. Beth, do you want to introduce the next speaker? And
with pleasure. So the last speaker in this session today is Myrte Faber, who's at the Donders Institute for Brain, Cognition and Behavior in the Netherlands. So Myrte's very, very interested in language and semantic memory and higher order cognition, and also the neural underpinnings of discourse processing and mind-wandering behavior. And she's going to talk today about functional gradients in the anterior temporal lobes, one of my favorite brain regions. So thank you very much for joining us today.
Yeah, thank you very much for having me. So my name is Myrte Faber, and I will be talking today about how overlapping connectivity gradients underlie functional multiplicity in the anterior temporal lobe. And I would first like to point out that this work is not just my own, but was actually undertaken by a fantastic team of people at the Donders Institute, including Christian Beckman and Koen Haak as PIs, Isabella Chestik as the grad student, and Guillen Fernandez as a local collaborator. So I'll be talking, as Beth just said, about language and mainly about semantic cognition. So our ability to use and to understand language critically relies on the fact that we have something called semantic representations. So for instance, if I say a word like piano, then hopefully all of you will have some sort of representation of a piano. So some of you might have a more visual representation. For some of you, it might be more abstract. But what is important for understanding language, and especially for understanding language in context, is that you have an ability to select the relevant features of this representation. So for instance, if I'm going to tell you that I will be playing a piano, different features are relevant from when I tell you that I would like you to move my piano. And this is what is also referred to as controlled semantic cognition. And we know that the anterior temporal lobe plays a critical role in binding these features into coherent concepts and in selecting those relevant features for the context at hand. So that anterior temporal lobe is thought to serve as a so-called transmodal hub that is connected with these modality-specific spokes. So for instance, we know that the anterior temporal lobe displays connectivity with visual cortex, with somatosensory cortex, and so on. So rather than being the area that actually represents those individual features of concepts, it serves as that kind of hub that actually receives that modality-specific input and mediates it. And for this, it also needs to be integrated in a network of semantic control regions, which is represented by its connectivity with, for instance, frontal and parietal control regions. Now, these functions of the anterior temporal lobe appear to not be limited to the single word level, but they seem to extend to larger pieces of discourse as well, such as, for instance, sentences and stories. So previous work has found that the anterior temporal lobe is more active during sentence and discourse processing than during single word processing. So what this suggests is that besides integrating these features into a single concept, it also plays a role in more combinatorial processing of semantics. So integration across features to make meaning of concepts and across concepts to make meaning of discourse. Now, an open question is how the how those multiple functions of the anterior temporal lobe are actually implemented in this region. And one thing that we know from both diffusion tensor imaging and from functional connectivity studies is that the connectivity of the anterior temporal lobe is graded. So what that means is that instead of having these discrete subregions in terms of connectivity, there are these gradual changes in connectivity throughout the region, which we might be able to describe in terms of overlapping modes of connectivity. So let me explain what I mean by that. I know that you're all gradient folks, so you probably already know this, but I always find it easiest to think of this conceptually in terms of an analogy with region V1, so with primary visual cortex. And what we know is that in V1, there is this gradual shift in receptive field across neurons in V1. And we also know that V1 contains these two overlapping functional modes, right? So one that tracks the distance from the center of the visual field, also known as eccentricity, and one that tracks the angle around the center, also known as polar angle. Now, it is possible that describing a similarly graded area like the anterior temporal lobe in terms of these overlapping modes of connectivity change similarly provides a biologically and functionally meaningful description of the organization of the area. And so we can get at this by firstly doing a spatial decomposition of the area's connectivity to find its potentially overlapping connectivity modes. And fortunately, Koen Haak, who I think is also on this call, has developed a method that does just that. So the idea is that we want to obtain individual level maps of functional connectivity organization, and that we can obtain these maps using a method called connectopic mapping. So the idea is that first, for a region of interest, so in our case, we're interested in the anterior temporal lobe, you build a similarity matrix of the vertex-wise resting state connectivity with the rest of the brain, which is then submitted to a manifold learning algorithm called Laplacian eigenmaps. And I'm pretty sure you already heard about that today. For those of you who haven't, the easiest way to conceptualize this or to think of it basically is as a nonlinear alternative to, for instance, principal component analysis. So it is a data-driven way of disentangling the dominant modes of connectivity change, basically doing that spatial decomposition that I showed on the previous slide. So the output of this procedure are individual maps of connectivity change. And importantly, we can summarize these maps in terms of their spatial layout using a spatial statistics approach called trend surface modeling. And what that does is that it fits a number of polynomial basis functions to describe that map on the individual level. And these parameters of those polynomial basis functions can be used to test for functionally relevant relationships with behavior. And in this case, we applied the connectopic mapping approach to the Human Connectome project dataset, from which we used resting state data for 766 participants to derive for each individual the first two dominant modes of functional connectivity change. And we then summarized the spatial layout of each of these, of each individual's anterior temporal lobe connectivity profiles in a few spatial summary parameters. And we then tested whether these at the individual level are predictive of behavior. And fortunately for us, there are two tasks in the Human Connectome project dataset that are actually relevant for that validation here. Namely, we have a picture vocabulary task and there's a story comprehension task. So the picture vocabulary task is a task of the standard NIH toolbox, and it consists of people hearing a word and then seeing these 4 pictures and then having to pick the picture that actually corresponds to the word. And for this task, we took a simple accuracy measure as our measure of interest. There's also a story comprehension task, which is part of the binder task. And it consists of people listening to brief stories and then answering a 2 alternative forced choice question about the story. So in this case, for instance, whether the story is about revenge or about reciprocity. And we take a measure that accounts for the speed and the accuracy with which people answer these questions as our measure of interest. And what we find is that those first 2 dominant modes are organized such that the first mode goes from superior to inferior. We see a second one that goes from anterior to posterior. Now, as I explained, we can use that set of polynomial basis functions. We can fit those to each individual's map to describe them in a small number of parameters. And here we do that on the surface. And we can use these to look at behavioral associations. So when we look at behavioral associations between these gradients and the tasks that I just mentioned, in this case in the left hemisphere, we find that that first gradient, so superior to inferior, is linked to individual level story comprehension, but not to picture vocabulary task performance. Now, in contrast, that second gradient, so anterior to posterior, is related to performance on the picture vocabulary task, but you probably guessed it, not to the story comprehension task. So as a control task, we also looked at whether either is predictive of performance on a math task, and neither is. So it seems to be specific to these tasks that require semantic cognition. We also did the same analyses for the right hemisphere and found no such associations. So that seems to suggest that these meaningful behavioral associations are actually left lateralized. So an interesting question is, what do good and bad performers on this task actually look like? So what I'm plotting here are the top 10 and bottom 10 performers on each task and an average of their corresponding connectopic maps. And as you can, even just by visually inspecting these maps, as you can very easily see, there are clear differences in how that connectivity is actually distributed across the anterior temporal lobe. So what does that mean? Now, so to get a sense of what these gradients are actually getting at, and just for illustrative purposes, what I'm visualizing here are the extremes of each gradient and how those extremes are connected with the rest of the brain. Now, it's important, and you probably all know this as gradient folks, that this is a pretty suboptimal way of thinking about anterior temporal lobe connectivity, given that we have these overlapping modes of connectivity. So taking an extreme does not account for that. But it can at least give us a bit of a sense of what these modes of change are that are represented in these gradients that we're looking at. And as you can see, for that superior to inferior network, we go from what looks like an extended language network plus some of the modality-specific spokes to frontoparietal control network that has previously been linked to language-related tasks such as reading and semantics, in addition to executive control. Now, if we look at our anterior to posterior gradient, there seems to be a gradual shift there from what looks like a classical language network to the other half of that frontoparietal control network, which has previously been linked to decision-making, emotional processing, and mentalizing. So what this seems to suggest is that the organization of the anterior temporal lobe is such that we have these 2 overlapping modes of connectivity, each of which integrates language networks with control networks, and that this might enable the role of anterior temporal lobe in semantic cognition. So to select and to integrate features to make sense of concepts, and to select and integrate concepts to make sense of discourse. I would again like to thank the wonderful team and our generous funding. And for those of you who are interested, we have put a preprint online that you can look up, or you can always email me if you have any questions. Thank you very much.
Thank you for that lovely talk. Really beautiful work. Does anyone have any questions that they'd like to ask?
I could ask one, Myrtle. Yeah, thanks for the talk. That was really great. You know, we've also looked at the temporal lobe, and there's like just one thing that is like always coming up again, as you may know, possibly, is like distortions and SNR. What would be your recommendations to, you know, for acquisitions to run in the anterior temporal lobe, and then maybe also how to control for like SNR gradients that we have and B0 distortions that we get for like diffusion sequences, but like also fMRI, for example?
Yeah, that's a really great and really interesting question. I wish I had a good answer here. We did actually look at it in our data to make sure that our gradients were not driven by these types of distortions. Unfortunately, I'm not an MR physics person, so I don't have any direct recommendations for you, unfortunately. We all know what the issues are, especially when you go to the more inferior regions of anterior temporal lobe. So yeah, this is an issue. And in this data set, we actually checked whether there were any such distortions that could drive the gradients. And that was not the case. There's no behavioral, I think what we did was that we checked whether there were any associations between the distortions and the behaviors that we see, and there were none. So you cannot explain the relationship between the gradient and behavior by looking at the distortion, at least, which seems to suggest that there's something meaningful going on in these gradients.
Yeah, no, that's a good sanity check for sure. Thanks.
So we have one question in the chat. How did you choose the optimal numbers of the gradient?
So that's another great question. That's not a trivial thing, obviously. I think we all know that model selection, defining the number of models that you need to optimally explain all of the variance in your data, is an unsolved problem in this world, so we don't really know. But this was very strongly driven by actually some of Beth Jeffries' wonderful work on the anterior temporal lobe, looking at semantic dementia and looking at semantic aphasia and other disorders where there seems to be a strong dissociation between different clinical profiles and different language-related issues that people have. So vocabulary knowledge, for instance, seems to deteriorate when there's atrophy from the temporal pole going backwards, but also that the connectivity with the rest of the brain is very highly important for being able to select the right features to understand language. And that was actually one of the things that drove this project, at least for me, trying to figure out whether we can find that dissociation in terms of gradients that could actually explain the different clinical profiles that we see.
Fantastic. And there's one more question in the chat here. Very interesting results. How did you select the 4 seeds in the last slide? And did you conduct a seed to whole brain voxel-wise correlation?
So how did we select them? In a pretty unprincipled way, in the sense that we just took an anterior point, a posterior, or actually a small section of the anterior bit, posterior bit, etc. And it's just for illustrative purposes only, right? So it's just so that we can have a bit of an idea of what are the extremes of that gradient? Where does it seem to be shifting from, and where does it seem to be shifting to? So it's not a very principled way, unfortunately. And yes, that is, well, it's a seed to whole brain analysis.
Fantastic. One thing I noticed about the last slide is that your different patterns of connectivity that you get from the extreme ends of the gradient themselves actually are not always completely different, right? So although the gradients capture differences in the spatially across that area of cortex you're looking at, the patterns of connectivity you get from the ends of those gradients they're clearly not orthogonal or not unrelated to each other. And your interpretation, as far as I understood, was also not completely unrelated.
Yeah, I think what might be happening is that you have basically two fairly similar networks that are shifting in this way, right? We have 2 bits of a frontal-parietal control network where we're shifting from what I think Matt Dixon and others have identified as a frontal-parietal control network A and frontal-parietal control network B. So we seem to have this shift that in frontal-parietal control regions that are connected, which is also behaviorally meaningful. People have shown that using meta-analytic tools, for instance, that each of those parts of the network is actually linked to different functions. And similarly, we seem to have slightly different subsections of the classical language network. So one that includes only the core language regions and one that seems to include more of the like modality specific, but language related regions as well.
Yeah, fantastic. So Boris, should we open it up for a broader discussion then?
Yeah, I think that's a great idea. Thanks to all the speakers so far. So the audience, you can now ask your questions to all the speakers, either through the chat, or you can turn on your camera and microphone, hopefully, or raise your hands if you would like to ask a question. Maybe while we wait for people starting to ask questions, I have like one that's maybe, you know, to Myrto obviously also, but maybe to like all the speakers is like, how should we, like, if we can obviously do analysis at the group level, so take like a mean connectome or, you know, but how should we go about studying gradients at the individual subject level, and what's the issues there? Do we need to align the different decompositions that we get from different subjects, or should we rather have other strategies? You know, there's like this joint embedding approach from Ting Xu, for example, developed Procrustes alignments or no alignment at all. What is the general sense of how far do we have to align the different subjects, and how little do we have to align the different subjects? Because I think for like BBM, it's like a similar issue, for example, when you want to find group differences versus group level variability, that like sometimes the alignment can also create something that is maybe too rigid. So any sense here? What's your recommendations?
So I'll start just because I just gave the last talk, and you might still remember what I'm referring to when I'm talking about trans-surface modeling, right? So the approach that we take is one where we use a spatial statistics approach to quantify the organization of these gradients. So you have a set of polynomial basis functions, and you can use those to basically describe what the landscape looks like, right? So this is something that comes from geostatistics, for those of you who are not familiar with it. And people use it to, for instance, describe mountains, or you could describe like the moon surface with it, right? So you can use it to describe a crater, for instance. So you have these different polynomial basis functions. They have different parameters, and they describe things like the steepness, or whether your steepness, whether it's like a linear slope, or whether you have some sort of quadratic trend, etc. The benefit of doing that is that you have a set of parameters at the individual level. What is a bit difficult is how to interpret them directly and how to make direct comparisons across participants. And in that sense, I think the approaches by, for instance, Ting Xu might be more, well, they're different, but they can be, I think, very valuable for actually doing those one-on-one comparisons across participants, rather than having these very elegant measures that you can very easily regress against behavior, for instance, but might be less intuitive, at least for understanding individual differences at the gradient level.
Thank you. Some of the other panelists want to...
Maybe I could like add just one thing from our work. So like what we did, we also wanted to check these effects in on an individual level, and what we ended up doing was to use Procrustes rotations to basically align what we get from PCA to one of the subjects. So we didn't try to align it to the group level results, because that would probably be that could kind of induce some. So we wanted to be like maybe more conservative, but still trying to like align subjects to one another, because with PCA. So I would say the least maybe again, in the case of something like PCA analysis, we should sort of try to maybe at least align the signs of what we get with the weights, and because of the kind of the mathematical kind of method we are using with PCA, we could be sort of, so it's not guaranteed that we are looking at the same thing. So in order to be able to compare them, in our case, what we ended up using was progressive rotation, which worked pretty well. So yeah, I would say some alignment is probably necessary if you're comparing like maps and these lower dimensional representations of the data. But we don't want to kind of overdo it, right? We want to, so yeah, that's what we ended up doing in our case.
So I can see we have a question from Suzanne Eisenhower. Would you like to unmute yourself, Suzanne, so we can hear you?
Yes, thank you. So I have a question for Katharina. I was wondering whether you could elaborate a bit more on the advantages of the harmonic modes in contrast to functional connectivity, but also the other way around, whether there might be any advantages of functional connectivity for EEG or MEG data.
Yes, thank you for your question. So I would say, first of all, the biggest advantage of using this approach that I showed, where you use the harmonic modes or building blocks extracted from the structural connectivity, is like the reason that we used that was almost purely pragmatic, because as was just mentioned, otherwise you kind of have to align across subjects and all of that. So we just used a group connectome, and then we were able to use the same building blocks for all subjects. And it was much easier than first trying to decide how to compute functional connectivity and all of that. And I think if you want to analyze what's going on in a task and you want to kind of model your data in first approximation as having something to do with linear combinations of networks, then this is an okay approach. But here comes the disadvantage, and why using functional connectivity might be nice is, of course, that as was also already mentioned in some questions, the connectomes, the structural connectomes that we use have many shortcomings, and they're not expected to align fully with the functional connectivity, of course. But even if we had perfect structural connectomes, but we don't. So for example, when you look at the harmonic modes that you get from the structural connectomes, some of them don't make sense for functional connectivity. Like you have the missing interhemispheric connection, so you have a very strong left versus right component that doesn't really play that big of a role in the functional connectivity. So yeah, there would definitely also be an advantage of using the functional connectivity if you can kind of, yeah, take an average or find the best connectivity measure to use in this context. Does that answer your question? I'm just going to take this as yes.
There's definitely nodding. Fantastic.
Okay, great. Yeah. Sorry, I don't see any videos. So,
So I've got a question that's kind of for everybody, but it was particularly inspired by Kevin's talk. So we've seen different gradients over the last two sessions, in fact. So we've got some gradients which resemble the Margulies hierarchy from sensory motor cortex to heteromodal cortex. And we've seen other gradients which look more like a kind of anterior-posterior distinction, which obviously is going to spatially overlap with the Margulies hierarchy in the temporal lobe in particular, but then looks different in other places. When I was looking at Kevin's data, I started to wonder whether, you know, if the hierarchy gradient might be present in the data if you removed the sort of global spatial kind of pattern. So there was a hint, I think, that the very most anterior part of prefrontal cortex, for example, was maximally different from motor cortex, although that difference was kind of swamped by the huge difference across the whole brain. So vision being very different from the premotor cortex, prefrontal cortex, sorry. So that's a kind of specific question. And then, you know, now we've got local gradients that Myrta was talking about, which capture the functional change within, in this case, a region of the brain which is quite near the top of the hierarchy that's described by Daniel's first gradient. You know, so we've got a question about how those local gradients might relate to global gradients. If they do, you know, are these local gradients maybe kind of capturing aspects of global gradients that you can see in these local areas, or is it like a combination of different patterns of connectivity change which reflect functional change in the anterior temporal lobes? I'm just curious to know how we relate these different observations together. Kevin, do you want to start, since I picked on your...
Yes, sure. Actually, that's what we have analyzed in our paper, but I didn't put in my slides. So we looked at some local gradients along the visual pathways, auditory pathways, and try to see how the gradient changes, and global gradients. And then, so how we describe it. So there are some global gradients and some local gradients that are how to say it, building this global gradient. That's my impression. And about your point that we remove location from peak frequency to see how it changes over hierarchies. Yeah, that was an interesting thing, because the main point was to see how peak frequency changes over hierarchies. But then the question comes up, how you define hierarchies? There are many ways to define hierarchies. And what we used was cortical thickness, which to see how peak frequency changes. But of course, there could be many other hierarchical, many other methods to define hierarchy from brain area to another area. And yes, but overall, I also observed this thing that there are some global patterns and some local patterns in the data. But I think for global patterns, ideas mostly come from structural analysis. Because from structural analysis, we know some gradients. But for local, I think they should be maybe more functional, specific gradients. Yeah. I hope this adds something.
Fascinating. Anyone else want to chip in?
I think there's...
yeah,
okay.
Sorry.
No, please.
Continue.
Chip in first, yeah.
Well, maybe you want to finish your thought first.
No, I would have gone to like a different question, actually. So please chip in.
Oh, okay. Yeah, okay, great. So, yeah, so I want to say that definitely there are many gradients. And this question of hierarchy, I would say we've heard about kind of spatial hierarchy in different ways, also functional hierarchy, and also this temporal/frequency hierarchy. And I think there's definitely a relationship between all of these, as it has also been mentioned that as you progress through the hierarchies, through the functional hierarchy, you get different time scales. And we also know, for example, that if you have low frequency oscillations, they tend to be more global spatially. So my kind of not very informative answer is that I think that these gradients all exist at the same time, and it has to do with this multiple functionality thing that was presented at the end. Like each brain region has many different functions on different temporal and spatial scales. So for example, if you would look at something like this is one result that we got from where we looked at functional connectivity of fMRI at a very high resolution. And if you look, for example, at a hand area, like a somatotopic hand area, then in the first two gradients, you can see it fully integrated in the somatomotor network. So it's really embedded in this global gradient. And what this means is that then the functional connectivity of this hand area is on this scale, from this point of view of this first or second gradient, the functional connectivity is very similar across the entire somatosensory network. And it's in opposition to like the visual network, let's say. But as you go to higher order gradients, you can see that the hand area can kind of disconnect, let's say, from other areas. And in one of the gradients, we saw an amazing opposition between the left and the hand and the right hand areas, where they were at opposite ends of a gradient that was spatially much more local. And so the hand can be embedded in motor function on a more global level, but left and right hand can also do completely different things. So yeah, I think this really integrates kind of all of the talks that we heard. So yeah, I'm a big fan of this topic.
Yeah, I'll add a very brief note to that, which is, I mean, I just explained all of this, right? So how in my local gradients that, as Beth just suggested, are at the top of Daniel's hierarchy, basically. What you see are these shifts from these modality specific regions to these heteromodal control regions, right? So you see that same kind of shift even at the connectivity of a local, like even at the local connectivity level, which might be a canonical motive in the brain, right? All of these like gradients, or many of these gradients at least, seem to have such an opposition, which I think might be beneficial, as I suggested in my talk, for exactly the integration of information.
Thank you very much. I can see that Casey's got her hand up. Do you want to chip in, Casey?
Yes, I had a question, if that's okay. It's kind of a general question because you all use quite different techniques, I found, which is really nice and complementary part of this session. So maybe that will come into this question of, do you think that information flows along the gradients that you've shown? And maybe that's both theory driven, like what you believe these gradients are showing, and also maybe whether the technique you use is sensitive to information flow. Anyone can go for it. It's very general.
What a great question. Who's brave enough to answer first?
So maybe I could like very briefly just answer that question. So that's a great question, actually. For example, in our case, we didn't really directly look at it, but in our lab, our postdoc Bertha, they have like a paper where they look at communication patterns, and they try to kind of see if the communication patterns following shortest paths, do they follow unimodal, transmodal hierarchies? And the answer is probably yes, to some extent. So it's kind of like if you follow a signal from a source to a target signal, what happens is usually they sort of, depending on, of course, like the start and the source and the where the signal is going, basically, but mainly there is also this component that's like kind of following the hierarchy. So if you are in a lower order area, you are probably, like as a signal, you are probably going to stay around the same area. But then if you are going to go to another level of processing, you would probably follow the hierarchy. So maybe that's something kind of to think about. Maybe it could answer your question a bit, but yeah. But that's what they've sort of seen. So looking at functional hierarchies and comparing it to structural sort of connectivity and pathways in structural connectivity.
Anyone else want to give it a go? Or Boris or Beth, what are your 2 cents?
I guess in Katharina's talk, there was a sense of the progression of processing types, which makes sense in a hierarchy, sort of proceeding in a particular sequence, right? Is that, did I interpret that correctly?
You mean because I showed how the different combinations of harmonic modes were activated along the course of the processing of the stimulus? Or what did you mean? Yeah.
Sorry, there's a lot of nodding and we forget to say it out loud.
Okay, yeah, sorry. I would say that that's a different question from if the information flows along the gradients. Because that would be maybe more like how they are, how the information flows between the different gradients, what does it even mean when I say that they activate at the same time, or they overlap, or there's a linear combination of them? I mean, I can't answer that question. But we wanted to actually include something about information flow in our other paper about the fMRI, because I think it makes a lot of sense to speculate that when you have these gradients and you have a gradient from visual system to somatomotor system, then you're tempted to kind of do this perception to action process. But we haven't explicitly shown it, but I'm going to be brave and say I believe in this, and I think that we should show it more explicitly.
Yeah.
Yeah,
Yeah,
maybe just a quick comment on this is you know, as you know, like none of the methods is like perfect, right? And mapping something like information flow is possibly quite complicated to do. But you remember maybe like from Seok-jun's beautiful talk in the first session, at least this convergence between the primary Magoulian gradient and the stepwise connectivity as being like, you know, it's like resting state fMRI, static connectivity. So it has to be taken with like a grain of salt. But obviously there's like this, at least this convergence between the steps in connectivity space and the principal gradient. And then there's like also this work from Bo-young Park, where he basically used hidden Markov models as being something that gets like a little bit closer to dynamics, and then correlating the patterns of the functional transitions at the group level, so not single subject, but group level, with these diffusion gradients. And at least, you know, there is like some association, at least between the structurally determined hierarchies and the way that the functional signals change. But as you also know, you know, it becomes like a little bit more challenging when you go towards higher order areas, because then maybe things become more parallelized, and it's like not so serially determined by a hierarchical model of cortical organization, because there's just something different happens. And I don't know whether we have like a good way to, at this point, really establish the communication flow that happens in like the DMN, for example. But I think like work that you presented,
really good question,
is you know, important for this.
Yeah.
So.
Yeah, I think that we all share this common theme that we're very good at predicting this like centrifugal, like sensory transmodal flow. But it just makes me wonder more and more about what's happening in the association cortex, where it isn't so easy to explain it by these, well, I only really believe in the centrifugal hierarchy, I guess, in terms of what's a hierarchy, but what all the other connectivity is doing and how we can model that as well. I think that so many of our techniques are sensitive to that very prominent one that comes out of visual cortex, but the association cortex, I think, is it's a big challenge for all of us. But it's nice to see many different techniques being used here.
Yeah, cool. I have maybe like one question, because, you know, I think Casey's question was like, would have been like a nice broad wrap up question, but we still have like 5 minutes on the clock. So I'll ask just something like on pre-processing and, you know, data preparation. And obviously there's like no general answer to this, but just, you know, to get like a general sense, like, and how far is the solution that you get from like these gradient, you know, connectome level decompositions somehow depending on what we do initially to the data in terms of spatial alignment? So like one question, it just came up like during Keivan's talk also. I was like surprised that, or at least, you know, I've never used MEC, I've never studied MEC data, but I always thought that there's like some dependence with sarcogyral folding, for example, and, you know, things that are like just maybe basic cortical anatomy. And how far do you think we have to correct our data for like these features, or should we just let the data talk?
So I think in case of MEG or EEG source constructed data, this problem, this pre-processing becomes even more important, because on source constructed data, you know, there could be frequency leakage. For instance, you can imagine one source in the posterior visual area, another source on frontal area, then their interaction would create some spurious gradients. So one should take care of all those spurious gradients. So the pre-processing then becomes very important. And another thing is that we in our analysis, we looked at peak frequency and we checked gradients, linear gradients, which is defined by slope and offset. So any factor that could change a slope or factor then would be an artifact to our gradient. And in our case, it was 1OF component was one thing, and any changes in 1OF component or its properties could change our gradient. And then what we will get out of our analysis would be a clear gradient, but that's actually artifact. So I think that's very important point to be careful when one applies gradient-based analysis. But another thing could be about gradients that are not linear. I've not done such analysis, but I can imagine that there could be such non-linear gradients. So, and I have no idea that how one can analyze them or put them in fitting models.
So I would say when you use graph Laplacian, that's nonlinear at some point because of the way that you prepare your graph Laplacian. And I would definitely, I fully agree with what you just said, that the pre-processing is very important with MEG and EEG. So I would say there we have to take care to relate gradients to behavior if possible. So that is always very strong support, I would say. And yeah, I would even add on top of that, that the way that you obtain the gradient also has some parameters, like the number of nearest neighbors that you consider or stuff like that. As before, we also had this question about how to set the parameters in the diffusion embedding. So we also found, playing around with this a bit, that the first few gradients are quite robust. But then when you get to higher order ones, which are more interesting, which is the challenge of the association cortex, then it becomes extremely fishy, let's say. So yeah, a lot of optimization to do.
Yeah, and I think another thing could be to look at individual level, to see how they look at individual level, then go to group level. And in our model, we use linear mixed effect model, which combine both group and individual level. And I think it helped a lot, at least in our work.
Yeah, I think Katharina already made the point that I wanted to make, but from an fMRI perspective. So with fMRI, you obviously have a degree of spatial smoothing, right? So if you smooth your data enough, you'll always end up with some sort of gradients, basically. In our study, we used very minimal spatial smoothing we used a minimal processing pipeline from the Glasser paper that I think most people who use resting state data will be familiar with. And what Katharina just already suggested is that I think the most valuable step here is actually the validation against behavior, like we did here, which was, as I explained earlier, a theoretically driven decision, trying to disentangle these distinct aspects of semantic cognition and actually trying to see if you can capture these meaningful differences at an individual level. And I think that that's a very important step in understanding those gradual patterns of organization.
Yeah, I think that was like a great session and a great discussion. So I would like to thank all the panelists for the excellent talks and the really thought-provoking points that they made. And I would like to thank my co-chair, Beth, for doing this. And I'll pass over to Casey, I guess.
Yeah, thank you. I really enjoyed that session. And thank you so much, Boris and Beth, for handling everything and to all our speakers. So in the next minute, I want to quickly give you the answers to the trivia. So let's do this. We've been running this workshop for 2 years. Okay, my PowerPoint's warming up. Give it a second. Here we go. To get a gradient, you do some type of dimensionality reduction on a covariance matrix. So dimensionality reduction, PCA, diffusion map embedding, happy with all those answers. From sensation to cognition is Marcel Meslem's opus, in my opinion. Brodmann had his habilitation thesis rejected by the University of Berlin. There's a really funny, well, it's not funny, but there's a story about the enmity between Paul Flessig and Oscar Fott. Read some papers by Carl Zillis on this, it's fascinating. Francis Gall, who many of you possibly know in relation to phrenology, in his day he called it organology, and he went on big tours of Europe promoting it. And the Fotts were the ones who said that the borders between areas are sharp as a hair. I would have taken either of them because, you know, I think that anything that came from one of them, they're probably both talking about. So I'm going to jump to the last ones here, the multiple choice. The ends of the dual origins are olfactory cortex and hippocampus. So we just had olfactory there. The platypus does not have a corpus callosum. Out of all the mammals, the ones that don't have a corpus callosum are the monotremes and also many marsupials as well. So if you notice that there was only one monotreme in there, you would have got a point. Good biology. Okay, and then last one of that section was the natural coordinate system of the CNS was based on development. So it's based on the ontogeny of the brain and where the positions are at the level of the neural plate. Okay, so this one was, I think, the most commonly correct answer. A lot of people just wrote Daniel 2016. So I think everyone knew that that was the Margulian gradient. Then this one, I think only one person got. It's Bailey and von Bonner, 1951, the isocortex of man. I think it's one of my favorite neuroanatomy books. I have a signed version at home. But okay, then that last one. Oh, wait. I'm not going to show that one. So can you guys remember there was a white picture with, it was black and white and it showed some gradients. Who did the quiz and knew the answer to this? I'm looking for an honesty system because if you may have noticed, there was a slight error on my part in not asking people their names in the quiz. So there was one person who really hit it out of the park. It was Sean. Okay, Sean was the only person who knew that this was Sinides 1962, and he did really, really well in the quiz. So Sean is our is crowned the gradient king for the year. So thank you everyone for joining. That was really fun, and a lot of people did okay, but yeah, Sean, you really showed your knowledge of neuroanatomy there. Okay, so thank you everyone for playing, and we're going to now have a break until I believe 12:30, right? So we have a 40-minute break now. Oh, at 12:20, I think. Let me just double check. I think, okay, I'll believe that. Maybe around 12:20, we restart. I feel like it's meant to be 12:30. Oh, if you come back at 12:20, there's a yoga session you can do at your table, but the actual proceedings restart at 12:30. So if you want to take the full 40 minutes, that's also okay. All good? Okay, see you all back here soon. Hi, everyone. I hope you enjoyed that little yoga break. I really liked that, honestly. And now I'm just going to pass it on to Sophie and Richard. You want to take over the session?
Yeah.
Yes.
Richard, do you want to start?
Should we introduce ourselves first a little bit, or people obviously know who we are?
We can do this in silence, like from this yoga mindset. People can just see us and perceive us, the humans that we all are. I guess that should be enough. But Richard, shall I introduce you and you me, or?
Yeah, sure. That will work.
So this is Richard. He's from Rotterdam. He studied philosophy, and I didn't know this until a month ago, or maybe 3 weeks ago. But then he realized that also the brain is maybe the seat of consciousness or cognition. Anyway, he started in Utrecht to study the brain and neuroscience, and then moved to Cambridge to do a PhD. He was happy there and productive, and finding good collaborators within Cambridge and beyond. And so he finished his PhD where he focused on brain structure and looking at also autism and social skills to a postdoc, and now he's a research fellow. And recently he also had an amazing preprint on brain charts of the brain, which is a very ambitious and interesting project to hype it a bit more. And behind him is a painting actually of neurons, I believe.
It's a very impressionist interpretation, I guess, of neurons. So thanks for the introduction, Sophie. So this is probably a coincidence, but I think we're both from the Netherlands. So Sophie's also from the Netherlands.
This is not a coincidence.
This is not a coincidence, maybe not. And we should have probably overlapped at some stage, because we've both been in Amsterdam and Utrecht, and we both kind of spent part of our early formative years there, I guess. But Sophie is now a group leader at MPI, where she's doing some amazing work on gradients, and that's where our interest first started intersecting. And obviously, with all of the other wonderful people that are organizing this workshop, we've had some very productive collaborations over the years. So it's up to us to, I guess, lead this Evo-Devo session, of which I have to say Sophie's much more of an expert than I am, because I mainly do a little bit of work on development, not so much on evolution. So Sophie will know a lot more about that. Should I just go ahead and should we just start with the first talk? I can introduce the first speaker, perhaps, or do you want to say something more about the session, Sophie?
No, I think it is more than perfect. Yeah, start with the first. Will you also want to tell the fun facts about the speakers, like per speaker? Or?
So we asked all of our speakers, because this is the Evo-Devo session, whether they had a favorite dinosaur and what their favorite period of development was. And Ludovico, who's our first speaker up, told us that the Pterodactylus is his favorite dinosaur, because it's just the idea of flying dinos is amazing. And I completely agree with that. And I actually told Sophie this as well. And then she corrected me, saying that apparently Pterodactylus is not a dinosaur, it's a flying reptile. So we both stand corrected. Now we have to find another favorite dinosaur, I guess. And he also said your favorite part of development is adolescence, which is, I think, an answer that we've heard before. So maybe you can get into that, why that's your favorite period in development later. Just to further introduce Ludovico, he did his master's in psychology in neuroinformatics at the University of Zurich before he joined the functional neuroimaging lab at IIT with Alessandro Gossi in Italy, where he is now a PhD student, and his PhD project aims at unraveling the elusive relationship between brain structure and function, trying to understand how it underlies the establishment of brain connectopathies. And I guess that's what his talk is going to be a little bit about as well. So over to you, Ludovico.
Okay. So, hi everybody. I'm gonna share the slide. Okay. Do you see the slides? I take this as a yes. So, hi everybody, and thanks a lot for having me here. I'm super excited for this opportunity. So, as you see from the title today, I'm going to talk about gradients of functional and structural connectivity in the mouse brain. And of course, our story starts with the work of Margulies and colleagues, who were, at least to my knowledge, the first to show that the macaque monkey also exhibit this principal gradient, raising the hypothesis that cortical gradients might be an evolutionarily conserved phenomenon. As you can see here, we can clearly see that the macaque and the human, macaques and humans are evolutionarily speaking quite close. And what is not clear, however, is to what extent this organizational principle can be generalized across evolution. So, for example, it's not clear whether other species important for system neuroscience, such as the mouse brain, actually exhibit a similar organization. And to address these questions, we used a resting state fMRI, mapping gradients exactly as described in the Margulies' work. And what we found is actually, in my opinion, super interesting and exciting set of results. Why? Because we found a first gradient that we labeled gradient A that was anchored at the one end in transmodal brain regions, and at the other end in unimodal brain regions. And the transmodal end of the gradient is what we call the mouse deformal network, whereas the unimodal part of the gradient is what we call the laterocortical network of the mouse brain. Then we also found a modality-specific gradient that was peaking on the one end in auditory brain regions and on the other end in somatosensory brain regions. So this actually is exciting and interesting for us because it shows that the mouse brain, to some extent, recapitulates organizational principle actually that we have seen in higher species. And in fact, our results are quite in line with analogous investigation of cortical gradients organization in the mouse brain. Here, I would just like to briefly introduce two excellent works. So the first one from Fulcher and colleagues. Here, the authors use T1 to T2 weighted imaging, and while their spatial resolution was not at the voxel level, they were somehow able to recover this polymodal unimodal segregation. And what is perhaps even more interesting is that they were able to relate this principal axis of connectivity variation to actually a number of different microstructural properties of the mouse brain, ranging from seat architectural type to axonal connectivity, for example. The second work that I would like to introduce was published recently by Unterburg and colleagues. And here, the authors, once again, using resting state fMRI, found a similar principal gradient, similar to the one that we described. And what is interesting here is that the authors were able to relate the gradient expression to neocortical evolution. So taken together, I say here that these results actually increase our confidence in the validity and generalization of these phenomena. And in fact, getting back to our original question, we argued that this unimodal segregation is the organizational principle that is evolutionarily conserved. So here on the left, I tried to put together a tentative diagram illustrating sort of the cross-species similarities in the deformal network. And at the same time, even though we argue that this unimodal polymodal segregation is evolutionarily conserved, we are also well aware of some species-specific cortical arrangement, which I think can be beautiful seen here from the scatter plots on the right. And in fact, so far we talked about resting state fMRI, but we actually had a number of burning questions. And to start with, the very first one was about the relationship between structure and function. So to be more specific, so what are the structural bases of these resting state connectivity gradients? And to address this question, of course, we took advantage of the amazing work of the Allen Brain Institute, who recently released a voxel scale model of the mouse connectome. So here we are talking about viral tracing experiments, in which we actually have even the potential to reconstruct the connectivity between single neurons by taking into account directionality. What we did was to perform a bit of downsampling. So this is technicalities that I'm not going to cover here today, but that you can find in our in the Science Advances paper that we just published. But the net result of this downsampling procedure is that we had the unique opportunity to map structural gradients basically at the voxel level. And what we found was, again, to me, super exciting and interesting, because we found that structural and functional gradients actually exhibit similar topology, especially if you compare gradient A across structure and function, they are virtually identical. And why is this important and interesting to us? Because first of all, we found a sort of very intimate link between structure and function, but also between different levels of brain organization, because structural connectivity was mapped basically at the mesoscale level, whereas functional connectivity at the macroscale level. And given this information, we also tried to extend. We wondered whether it was possible to extend this relationship to map not only to find not only a link between structure and function, but also to resting state fMRI dynamics. And to map dynamics in the mouse brain, we used co-activation patterns. This is again work from our lab. Co-activation pattern consists in a simple frame-wise clustering procedure in which we basically try to map the ups and downs of the ball signal. And what we found here is that a very limited number of fMRI states or co-activation patterns actually account for the vast majority, for example, of functional connectivity. Raising the suggesting that this sort of the resting state activity is actually governed by these recurrent building blocks of co-activation patterns. And what we did here was to here we asked ourselves, okay, maybe this sort of the spatial topography of these co-activation patterns is somehow constrained or shaped by the underlying axonal scaffold. And what we did here was to correlate the co-activation maps with our structural gradients. And what we found was that indeed the polymodal unimodal gradient was the one that was strongly associated with one of these states, suggesting that maybe the polymodal unimodal gradient also is constraining resting state fMRI dynamics. The very last question we wanted to address was on the relationship between gradients and laminar defined hierarchy, because both humans and macaques, it has been showed actually over and over that there is a strong association between gradients and this laminar defined hierarchy. And here, once again, we took advantage of the amazing work of the Allen Brain Institute, who recently released this cortico-cortical hierarchy based on the laminar projection patterns for the mouse brain. And we, again, correlate this index with our structural gradients. And what we found here is that in this case, it was the modality-specific gradients to be strongly associated with the gradients, with this cortico-cortical hierarchy, sorry. To sum up here, we argued that the mouse brain gradients recapitulate architectural principle observed in higher mammalian species. So we found that the polymodal unimodal structural gradient is actually associated with both macroscale resting state fMRI dynamics and the spatial arrangement of cortical function connectivity, whereas this modality-specific structural gradient, as I just said, just predicts laminar defined hierarchy. And why is this important? So besides the obvious interest in mapping commonalities and differences across species, this is important because, in our opinion, it paves sort of the way to use the mouse as a simple model organism to study, for example, neurodevelopmental disorders. And in fact, this is what I'm currently working on. These are different mutations that have been associated with autism. So these are mouse model for ASD. And what we found is that across these mouse models, we found that an aberrant cortical architecture. So it was sort of recurrent results, which, in our opinion, again, I would really conclude this presentation by saying that it's actually the identification of cortical gradients in rodents, allows for the investigation again allows for the investigation of a typical gradient organization in psychiatric disorder and especially in neurodevelopmental disorders. With this, I am done. So I would like to thank you for your attention, for inviting me here. And of course, a huge thanks goes to the lab. Without them, I wouldn't be here. And last but not least, a huge thanks goes also to our wonderful collaborators. Okay. And of course, I'm glad to take any questions.
Yeah, I think we have time for maybe 1 or 2 questions if there are any in the chat. If not, we can always come back to that at the end when we have the panel discussion session. So I'm not sure. I'm going to check the chat if there's any questions. I don't think there are. So I mean, I have some, but I can park them for the panel discussion as they might overlap with some of the other speakers as well. So let's go to the next speaker.
Yes. The next speaker is Christina Charvet. I hope I pronounced the last part of your name right. Her favorite dinosaurs is the Tyrannosaurus Rex because it's mighty. And she's also curious how the developmental mechanisms, what they were leading to such small arms in this species. And her favorite developmental stage is birth. And I think that's indeed, it's a fascinating answer that I could talk about with you for an hour with questions, but we will not do this because you will present something more interesting, namely your research. And a short bio about Professor Charvet is that she obtained her Bachelor of Arts in Psychology at UCLA before completing a PhD in Biological Sciences at the University of California, Irvine, where she studied evolution of developmental mechanisms generating variation in neural structures linked with the song systems in songbirds and parrots. Following, she was a postdoc at Cornell University with a 2-year stop at the Harvard Medical School and George Washington University. And currently, she's an assistant professor at the Center of Neuroscience and Department of Psychology at Delaware State University, part of the Delaware Neuroscience Center. She's also the curator of Translating Time, which is a website that enables researchers to find equivalent developmental time points across species, which is also a great resource and a fascinating project. So without further ado, thank you for being here and looking forward to your talk.
Well, I hope you can hear me and you can see some slides. I really wanted to thank you for having me. I've really been enjoying the day. And I remember being really excited when I first learned about there being a whole workshop devoted to gradients last year, because we've studied gradients for some time now. And so it's really exciting to see that gradients are a fast growing topic of study. So today I'm going to talk about past efforts focused on gradients of organization and how they evolve. And I'm then going to talk about how more recent work focused on the integration of neuroimaging with transcriptomics to study the evolution of the brain. So in my lab, we study brains of different species. Here you can see a wide variety of mammalian brains, and we compare these brains with those of the human brain to understand what features are unique to the human brain. And so our work is mostly focused on the evolution and development of cortical organization across mammals. When we take a look at the cortex, we see that it's composed of the white matter, as you can see here, highlighted with pathways highlighted with diffusion and martractography, as well as the gray matter, which contains neurons and glial cells. And I'm going to talk about our efforts linking information from the white matter and the gray matter to study cortical organization and evolution across mammals. So I'm going to spend some time talking about past efforts focused on the evolution of cytoarchitecture within the gray matter. And I'm going to talk about how linking cross-cortical variation in structure in adulthood and in development can serve to identify developmental bases of neural structures supporting sensory specializations. And I'm going to talk about the evolution of vision in primates. And then we're going to talk about more recent efforts where we're integrating information from the gray and the white matter to develop better tools to study circuits. And we're really excited to use those tools to study the evolution of the cortex. So one of the things that's really remarkable about primates is their good vision. Primates stand out compared with other mammals in having remarkable vision, and the neural structures supporting good vision stand out in primates. The visual cortex, as highlighted here, is unusual compared with other mammalian species. It's characterized by this unusually high density of neurons. And when we use immunohistochemical markers, as shown here, we see these very sharp borders between the primary visual cortex and other cortical areas. This is something that is quite unique to primates. So we were interested in investigating these neuron densities further across the cortex of different mammalian species. So we took some data produced by Collins and Cass, where they would basically flatten out the cortex as if you would a pizza. And they would have this technique where they could quantify neuron numbers across the cortical surface. So we evaluated those data, and we found interesting variation across the axes of the cortex. Notably, there was this very strong gradient in the number of neurons per unit of cortical surface area from the front to the back of the cortex, where you can see relatively low neuron numbers towards the front that systematically increase towards the back. And it's in the back that we find the primary visual cortex. And we looked at other mammalian species, and we looked at a broad range of mammals like manatees, and we found no such variation. And here are just two example rodent species that illustrate that there was very little variation in the number of neurons per unit of cortical surface area across the axes of the cortex. So this phenomenon here was something that was really specific to primates, and it wasn't observed in other placental mammals. And so we looked to the process of neurogenesis, which is the production of neurons, to understand how modifications to neurogenesis could give rise to the smooth variation in cytoarchitectural gradients that we see in primates, but not other mammals. And here's a schematic of neurogenesis shown, where you can see the number of proliferating cells in dark purple plotted against different fetal ages in a macaque, and neurons in light purple. And in the background here, what you see are sections taken through a macaque at different stages of development. And neurogenesis is an entire process that lasts multiple days, as exemplified here in the macaque cortex. Early in development, the cortex is nearly exclusively comprised of proliferating cells. As neurogenesis progresses, cells stop proliferating, they exit the proliferative pool and migrate radially towards the outer surface of the cortex. Towards the end of neurogenesis, the proliferative pool wanes and the cortex is made nearly exclusively of neurons. And we collected as much information as we could about this process over the course of development across the cortex and in different species to resolve how variation in neurogenetic schedules might give rise to the smooth variation in neuron numbers that we see in the primate cortex, but not in these other rodent species. And in summary, what we found was that neurogenesis was unusually extended in primates compared with rodents, even after we control for variation in development schedules. And I'll walk you through our model, which was published in PNAS a few years ago. And this is a schematic showing neurogenesis in a rodent species, where you can see over time, shown here as duration, neurons are produced, as you can see here on the z-axis. But in the rodent, there's very little variation from the front to the back of the cortex. And as a result, there's very little variation in the number of neurons across the axes of the rodent cortex. A different situation emerges in primates, which extend neurogenesis compared with rodents, even after controlling for variation in development schedules. And the consequence of extending neurogenesis is that more neurons are produced. But they're not just produced anywhere. There's variation from the front to the back of the cortex. Neurogenesis is unusually extended in the back of the cortex, and this is where we find most of the neurons. So we thought that it was interesting that it's by extending neuron production that smooth variation in cortical architecture emerges and the evolution of good vision arises in primates. So when we look throughout the visual system, we see a number of instances where sensory specializations appear really directly linked to these gradients. One example is where we see these sharp borders between V1 and other cortical areas are linked to the smooth variation in neuron numbers across the primate cortex. As another example, we find in the fovea this specialized structure responsible for good vision in the center of our visual field is linked to systematic increases in cone densities from the periphery to the center of our retina, and the highest densities in cones are located towards the fovea. So these observations suggest that we should focus on variation across the axes of the neural structure, both in adulthood and in development, to understand the developmental basis of phenotypic variation across mammals. So obviously there are gradients in cell populations, and there are gradients in structural and functional connectivity. And so we've been interested in investigating the evolution of connections with diffusion MR tractography as exemplified here. Now, diffusion MR tractography does have certain limitations, and the tractography can be uncertain. And this uncertainty becomes particularly problematic when we compare brains of different species, because the brains have various shapes, and so there might be these confounds that emerge. And so we've been really excited to adopt an integrative approach to study the evolution of connections. And what our efforts have focused on is integrating information from the white matter with the gray matter, as well as transcriptional information from bulk and single cells. Here you can see a Thiessen plot that classifies cell populations based on transcriptional information. Every dot represents a cell, and these dots forms what we call these clusters, and these clusters are color-coded and represent different cell populations as exemplified here in the cortex. And so our efforts have focused on integrating across these scales to transcriptionally define neurons that we see with diffusion MR tractography. And I'll walk you through a recent pipeline that we've used integrating this information to transcriptionally define the neurons that are projecting through the white matter. It's these neurons that we're seeing with the diffusion MR tractography. And so we have done this by testing for associations between white matter maturation and gene expression over the course of development, as shown here in humans. We've used myelination as an index of white matter maturation. We've done this iteratively. And here you can see summary statistics where we've tested for the association between gene expression and white matter maturation. Here you can see the negative log p-value from these test statistics are plotted against the slope of this linear model. Here, a higher value means the test statistics is increasingly significant. We control for multiple testing, and we only selected genes that positively associate with white matter maturation. So we selected these genes shown here in red, a total of 3,576 in this case. So we did this iteratively across the cortex, and here are the cortical areas we considered. So doing this, we came up with a list of genes that significantly and positively associate with white matter maturation. We then filtered the list of genes further with single cell RNA sequencing, where we only selected genes that were expressed in clusters associated with excitatory layer 2 through 3 neurons. Those are neurons that we would expect to project through the white matter. We excluded genes that were expressed by non-neuronal cells so that we would make sure that the genes are expressed only by neurons and not glial cells and astrocytes, to ensure that our list of markers would be associated with neurons that project through the white matter. We then ensured that our list of genes had good spatial expression patterns. So here we have in situ hybridization, where we see that one example gene is expressed by these pyramidal neurons. These are neurons we would expect to project through the white matter. And so doing this, we found a total of 250 markers. And these are genes that should be expressed by neurons that are projecting through the white matter, the neurons that we should be visualizing with diffusion MR tractography. So moving forward, we're really excited to integrate information from diffusion MR tractography with this list of genes. Here are a few example genes to study cross-cortical variation across mammals. We've started to do this where we've investigated the evolution of the frontal cortex, integrating this list of 250 genes with structural variation in adulthood and in development, but in the interest of time, I'm not going to go over that study. We do have a bioRxiv preprint if you're interested. So I've walked through some of our long-term and short-term efforts where we've studied gradients in organization, both in adulthood and in development. This work demonstrated that linking cross-cortical organization at different stages of life serves to identify the developmental basis of neural structures supporting sensory specializations like good vision in primates. And more recently, we focused our attention to integrating transcriptional and structural information to define neuronal populations that we plan to use in combination with diffusion MR tractography. And so we're really excited to move forward with this approach. So I'd like to thank a number of people who participated with this work, especially Emi Takahashi and André van der Koe from the Martino Center. I'd like to acknowledge the work of members in my lab and the funding from the NIH. And I'd like to thank you very much for your attention.
Yeah, thank you so much for really the amazing work. So applause. It's a bit lonely applause, but everybody would join you, I guess, if we were not behind our own desk. We have one question from Casey. I don't know if she wants to read it herself or.
Yeah, I can say it. I was just wondering whether you have tried to account for the influence of individual variation in relating white matter maturation to gene expression. I asked just because if you have any ideas, I think it's really challenging with the few number of transcriptomics subjects we have and the indirectness of the relationship. I think it's something we're all struggling to deal with. So if you have any ideas.
Yeah, so that's really a great question. We've actually focused our efforts on removing as much individual variation as possible, making sure that the list of markers is reproducibly identifiable across data sets. And it's just going to be hard to find those kinds of large scale data sets in different human individuals. But that's a great question. So we haven't focused our attention on individual variation.
They're going for reproducible markers, at least in the imaging data set's a good start, I think. Thanks. Yeah. It was really an awesome talk. I really liked the work.
Thank you.
Cool. Now, I think over to Richard, who will introduce the last speaker of this session. So Richard, over to you.
Again, I was going to say, like, amazing talk, and I also had some questions, but I'll save them for the panel discussion. I'm sure there's a couple of questions that cross these different talks as well. My honor to introduce our next speaker, Graham Baum, who did his PhD at UPenn, if I got that correct, with Danny Bassett and Tad Satterthwaite, and is currently a postdoc at Harvard in the lab of Leah Somerville, where he studies circuit level markers of brain maturation and how they are linked to individual differences in working memory, inhibitory control, and decision making. And we also asked Graham about his favorite dinosaur, and he said the Brachiosaurus, because they're large, slow, and peaceful dinos who munch on a lot of foliage. I think that's an excellent answer to that question, to be honest. And he also said that adolescence was the most exciting period in development because it has a double-edged sword of heightened plasticity and vulnerability, which again I think is a great answer and like very much one of the reasons I'm also interested in studying adolescence. So I might be a little bit biased on that. Without further ado, Graham, you want to start your talk and we can dive right in.
Great. Yeah, thanks so much for the introduction. I actually also originally thought that pterodactyl was my favorite dinosaur until I realized that it wasn't actually a dinosaur. Okay. Can everybody see everything okay?
Yeah.
Okay, great. Well, thanks so much to the organizers for inviting me. I'm really excited to share my work. And thank you all for being here. Today I'll be talking about this recent paper is focusing on the development of structure-function coupling and human brain networks during youth. And so we know that the human brain is organized into circuits that develop from childhood through adulthood to support complex cognition. Critical behaviors like self-control, decision-making, and these circuits are anchored by white matter pathways, which coordinate the brain activity necessary for cognition. However, there's little research existing to explain how white matter develops to support the specialized circuits underlying improved cognition during adolescence, which is this period of rapid brain development. And so my research leverages large neuroimaging and behavioral data sets to characterize how foundational processes of brain development from childhood through adulthood support these improvements in higher order cognitive ability. And usually I would devote a large portion of the talk to kind of motivating why I think adolescence is a really kind of critical period to study. It's been thought of as a sensitive period for neurocognitive development. And I think this figure captures this idea nicely, which is that 1 in 5 adolescents will develop a mental illness that persists through adulthood. And across the board, whether it's ADHD, mood disorders, psychosis, substance abuse, they're all linked to deficits in higher order cognitive ability and in abnormalities in brain development during this adolescent period. And so for me, this really shines the spotlight on this as an interesting period where there's lots of changes in brain structure and function and socio-emotional processing. And so it also poses this problem where adolescence is this period of heightened brain plasticity and vulnerability to developing mental illness. And so one solution, if you will, is to conduct this research to identify circuit level markers of brain maturation that track the development of higher order cognition. And the idea is that mapping the normative development of brain structure and connectivity, supporting healthy cognition, is a prerequisite for understanding how abnormal development processes could underpin the risk for mental illness. And so some of the questions motivating this study for me were, like, how does white matter connectivity underpin these coordinated fluctuations in neural activity during different cognitive computations, in this case, a working memory task? And second, does structure function coupling remodel during development to support executive function, this form of higher order cognitive ability? And this work was largely motivated by some older work by Packold and Rakesh and Dick Passingham, which pushed this idea of regional connectivity fingerprints being important for the functional specialization of a cortical area being determined by its unique pattern of anatomical connections. And so I illustrated this with a single subject from the data set I'll introduce shortly, where we can highlight an area and a frontal parietal area in the prefrontal cortex and map its anatomical connections, which we've reconstructed using diffusion tractography. And it gives us a sense of the distribution of connections to other networks and systems. This particular area seems to be strongly connected to default mode and motor networks, among others. And from that, you can make inferences about its role in cognition. And so to me, I was interested in expanding this framework of anatomical fingerprints to quantify the degree to which a cortical region's pattern of structural connectivity aligns with or predicts that region's pattern of functional connectivity. And so for me, structure function coupling describes these anatomical constraints on functional communication. And it occurs when a region's profile of interregional white matter connectivity predicts the strength of interregional functional connectivity. And so what are some different things that could influence variation in structure function coupling? What might this reflect in the brain? And so thinking about this from a developmental perspective, there's work over 100 years ago from Paul Flechsig that used, among others, that used histological methods and postmortem samples to show that early on, soon after birth, primary sensory cortex is heavily myelinated. And there's these areas distributed in frontal, parietal, and temporal cortex, which have a much more slow, protracted process of cortical myelination through adulthood. And the idea is that structure function coupling, in this case, could reflect the degree to which a region has become functionally specialized through activity-dependent myelination during development. So from this perspective, we predict that children early on would have high structure function coupling and primary sensory cortex, but later on through adolescence and adulthood, we would expect to see increases in these distributed transmodal areas in structure function coupling. And from a different neurobiological perspective, coupling could also reflect a brain's degree of functional specialization, ranging from these functionally specialized areas, unimodal areas that are specialized for specific sensory perception, you would expect these to have high structure function coupling to support very specific kinds of computations, versus areas that are responsible for more abstract and flexible computations. These areas highlighted in red, and we all know from Daniel Margulies' paper in 2016. And so again, we predicted that structure function coupling would vary across this kind of gradient of cortical specialization, which we defined in a couple of different ways. So we hypothesized that regional variability in coupling would reflect this gradient of specialization, and that age-related increases in structure function coupling would be localized in transmodal areas. These are areas that are supposed to support working memory and other higher order cognitive abilities. And they're also areas that undergo this prolonged period of myelination. And lastly, we want to test the hypothesis that individual differences in coupling during this working memory task would be associated with executive functioning outside the scanner. And the idea is that, beyond just being an important kind of element of brain organization, that structure function coupling might have implications for behavior as well, for cognitive ability. And so to test these hypotheses, I used data from the Philadelphia neurodevelopmental cohort. This is a really large data set. It started with roughly 10,000 children who were genotyped at the Children's Hospital in Philadelphia. And about 1,600 of them came back for multimodal imaging and cognitive batteries. And so my study reports data from a subset of this PNC sample who had high quality diffusion weighted imaging, fMRI, and structural imaging as well. And so we have 727 subjects between the ages of 8 and 22. To measure structural connectivity, we use probabilistic cartography and FSL to reconstruct these white matter bundles throughout the brain. And then using the Schaefer parcellation, we are able to quantify the strength of these reconstructed white matter pathways between each pair of regions. So we can try to understand, for example, the strength or the number of reconstructed pathways from this area in the visual network to all other regions in the brain. And that would be reflected as a row or a column in this connectivity matrix. And to measure functional connectivity, we did this during an NBAC working memory task where there's varying degrees of working memory load. We did a supplementary analysis with resting state as well and in different subsets of these task blocks. But overall, we just had more task data. So we had more reliable estimates of functional connectivity. And we wanted there to be kind of a cognitively relevant stimulus as well. But I can go into the supplementary stuff later on. But so using the same parcellation, we could derive a functional connectivity matrix during a working memory task, where the strength of functional connectivity is measured as the Pearson correlation in the mean regional bold time series. And so together, then we have for a given region, we have this vector describing its strength of the strength of connectivity from this region to all other regions in the brain. And we have the same thing for functional connectivity. And so I took a really simple approach to measuring structure function coupling by just taking the Spearman rank correlation between these vector elements from, say, region 400 in the structural connectivity matrix to region 400 in the functional connectivity matrix. There are many elegant, more complex approaches to measuring structure function relationships in the brain. And we decided to go for this relatively simple approach that gives us an estimate of coupling for each subject and for each brain region. And so when we average this across the entire brain, this estimate of structure function coupling, or I'm sorry, across all subjects, we can see that there are some cortical regions that tend to have higher structure function coupling. These are in somatomotor and auditory areas, and areas that tend to have low structure function coupling, particularly in temporal and parietal areas. And so we want to see if regional variability in coupling was related to other aspects of cortical organization. And we found that areas with higher structure function coupling tended to have more segregated patterns of structural and functional connectivity, meaning that they had stronger connectivity within their specialized networks. And we also found that areas with higher structure function coupling tended to be these unimodal areas responsible for sensory processing, whereas transmodal areas tended to have lower structure-function coupling. And finally, we wanted to see if this recapitulated a map of evolutionary cortical expansion from macaques to humans, where as brains get larger, these primary sensory areas tend to be highly conserved, whereas these multimodal areas tend to expand disproportionately. And what we found is that it's these highly conserved sensory areas which have strong structure-function coupling, and these newly expanded areas that are supposed to support potentially more complex cognitive computations, these tended to have lower structure-function coupling. And what we found in terms of age-related changes, these tended to be localized to these sort of transmodal areas in the default mode network. So these effects were enriched in the default mode network over and above other networks. And when we broke it down a little bit more, on the y-axis is kind of the magnitude of age-related change. And we found that these areas where structure-function coupling was increasing, they tended to be functionally segregated, so they have a low participation coefficient, yet they were also transmodal, which is a little bit counterintuitive. And they tended to be localized within this default mode network, which are the red dots compared to all other networks here in blue. And so we find that this is kind of localized in a specific part of the brain, mostly in default mode. We were able to replicate this using a longitudinal subset of the data, which is just reassuring that these age-related changes are actually reflecting intra-individual change. And lastly, we found that higher structure-function coupling in the rostrolateral prefrontal cortex mediated age-related improvements in executive function, again, suggesting that higher coupling in this area between structural and functional connectivity is actually important for supporting executive functioning measured outside the MRI. And so again, we found that structure-function coupling reflects these gradients of functional specialization, that age-related changes during adolescence were really localized to these functionally segregated yet transmodal areas of the default mode network, and that higher coupling in the prefrontal cortex supported improvements in executive performance. And so I think this suggests that similar measures of structure-function coupling could be interesting markers of plasticity and functional specialization in different contexts, both in learning different tasks over the course of the lifespan development, and also in different disease conditions. And so with that, I'd like to thank Ted and Danny, my PhD advisors, as well as a large team of people at Penn, the GERS who collected the PNC data, and Leah Somerville, my postdoc advisor at Harvard, as well, has been extremely supportive. And thank you all for listening.
Thanks for a great talk, Graham. I think we can dive in straight to questions or a panel session. I see there's a question in the chat. I'm not sure if the person who's asked it just wants to unmute and ask it directly themselves, if that's allowed, if that's possible.
I can read it. And
hello.
Yeah,
I guess this refers to the question I posted.
Yeah.
Yeah. So thank you very much for the great talk. It's very interesting and intriguing to see the integration of the functional and structural connectivity coupling together. So the question is how applicable do you consider the Schaefer Atlas for structural connectivity, considering that the Atlas grouped regions based on the underlying functional patterns? Do you think there will be a potential bias towards detecting stronger functional the aspects rather than the structural connections?
That's a great question, Peter. Thank you. And that's something that we kind of toiled with a lot when doing this study. And it's true that having a parcellation based on functional homogeneity and bode signal can influence, the networks and the measures of connectivity you can get. I would say that we felt like the Schaefer Atlas did a really nice job of both having cortical areas that with boundaries that made sense to us, but that also could be divvied up nicely into the networks, the intrinsic functional networks that we care about. But we also reran these analyses using kind of the standard, free surfer atlases, the Deskin atlas and things like that, where we found globally really similar results in terms of which areas had high versus low structure function coupling and where we saw the age-related changes. But within kind of internally within the group of co-authors, we decided that like the Deskin atlas and those sorts of anatomically grounded atlases don't do a great job of capturing the functional boundaries. So it was kind of a toss-up, and we decided just to replicate where we could with an alternative atlas. But if we had a surface-based pipeline or something like that, I would think that the the HCP multimodal parcellation would also be a good starting point with better resolution data.
Perfect. Thanks for the answer.
Sure.
I see Boris has his hand raised. So.
Yes, thank you all for the great talks and for the excellent session so far. This is a question to Dr. Baum. Thank you so much for the talk. It was really interesting. Just one point for clarification. So your functional connectivity is from the NBAC tasks. This is task-based, state-based functional connectivity. Would you expect the relationship to be seen also with different acquisitions like REST and or like a different task, or is this really something that's specific for the NBAC connectivity?
Yeah, excellent question. We think that, so I did this as well with resting state data, and I didn't have the opportunity to try it with other task contexts or states, functional states. But we found, again, very, very similar overall kind of spatial distribution of areas with high versus low coupling when we use resting state functional connectivity. The big difference was that we didn't see associations between regional structure function coupling and cognitive performance when we used resting state, whereas we did see this link when we used the working memory task. And there's also work, I think, from Todd Constable's group that shows that using task-based functional connectivity is more useful for predicting individual differences in behavior and that sort of thing. And so we decided in this case to use, or at least to focus our results with a task-based working memory task. But I could imagine there would be some subtle differences across different types of tasks as well. And it would be really interesting to look at those.
Can I just ask like one follow-up question? This may be unrelated, but you're possibly like familiar with the work from František, Vaza, with the conservative and the disruptive modes of the functional network changes in adolescence. And it's quite interesting that it spatially seems to co-localize a lot with the regions that show like this more disruptive pattern of connectivity changes, like those where you show the increase in structure function coupling, so in the higher order areas. In the Vaza paper, there was like a role for subcortical structures that shows like a similar change in connectivity as for the cortical areas, as for the higher order cortical areas. Is it something that you see there as well? Have you looked at subcortical nodes?
I have not looked at subcortical nodes. I think it's definitely worthwhile. There's a lot of interesting stuff going on, of course, in the subcortex. But that's a great question. And I definitely had read that paper, I think, back when it came out, but I don't remember the nuances between those different modes of functional development well enough to answer your question, I think. But that's definitely something I'd like to look into more.
Yeah, in that paper, it seems that the subcortical structures, they seem to mirror a lot of functional reconfigurations that also happen in transmodal areas.
Interesting. Very interesting.
I think we have a question from Sean next.
Hey, great, really interesting talk. So I guess my question is just a little bit about terminology. I mean, and you and also previous papers have talked about structure functional coupling or tethering, but really, I think it's just really one aspect of structure, right? I mean, there are other aspects of structure that vary. So for example, there was a paper from Rishi Chowdhury in 2015, I think I mentioned earlier, that suggested that some differences in differences between structure and functional connectivity may be explained by differences in the amount of dendritic spines and pyramidal cells, for example, which we think might be related to local recurrent excitation. So I guess, do you agree that maybe there are other aspects of structure that could better explain the functional connectivity patterns? And secondly, have you thought about any of these other, let's say, biological differences between regions that can better explain the functional patterns?
Yeah, that's a wonderful question. I think that there's a few things. I would certainly assume that there are microstructural properties, things like you said, related to spine morphology and density, and the sort of density of cortical myelin content as well, which could influence, at least on the structural side, the relationship between structure and function for a given area. I also think, so yeah, to answer your question briefly, I do think there's much more to it than just structural connectivity from diffusion weighted imaging and functional connectivity from a task. And on the functional side, I think there's also much more interesting kind of dynamics and spatiotemporal things going on. Like, we're just capturing the average correlation and the bullet signal over the whole course of the task. But you can imagine that there are kind of different time scales for functional communication and things that might not be captured by the simple measurement that we're using in this paper. And so I think there's ways to kind of expand on both sides. I would be really interested to think more about how to incorporate measures of microstructure and other things into the model. And I know there's a lot of people in this group who have done similar kinds of work, so I'd be interested to hear their thoughts as well. But that's a really great point, Sean. Thank you.
I know Casey has her hand up, but I might have a very short follow-up question to the question you just asked also about terminology, because I very much noticed that, Ludovico, in your talk, you were talking about like one being a constraint on the other. And I was just kind of curious whether it was a very conscious decision to talking about like structure kind of constraining function rather than talking about the association between the two, whether you can actually in those animal models might maybe even look at like some kind of more causal constraint if you're looking at it across evolution and development, perhaps. You're still muted, I think.
Okay, to me, so sorry, I go first. To me, at least, I would say yes, it's more structured than constrained functions, especially at least for what concerns the mouse brain. Actually, I would say so if I were to speculate. Then to what extent it's also sort of flexible association, it's some excellent questions. And we're actually also trying to tackle these kind of questions in the lab, for example, by using optogenetics and similar approaches.
Thanks. Casey, do you want to ask your question?
Yeah, you kind of asked my question, Richard. But yeah, I think that it's, well, yeah, I'll just ask a different question rather than like kind of banging on about structure, function, coupling, because I'm also of the opinion that we need to stop saying that they're uncoupling because, well, okay, maybe I'll just ask it anyway and be a little bit provocative, but how could it be that function isn't related to structure? Like, isn't it just that we're missing something in our model of structure? Maybe that's kind of what Sean was getting at without being so direct, but isn't it a failure of our model rather than uncoupling of structure from function?
To me, it could also depend from the data itself. For example, we used monosynaptic connectome in our work. So maybe for all the connections type that are polysynaptic are just lost. Yeah. For example, cortical connectivity in the mouse brain is mainly monosynaptic, essentially. For example, cortico to striatal connectivity could be, or striatum to striatum could even be polysynaptic, and this is of course then lost. Yeah. At least in mice models.
Yeah, I completely agree. It's more that there's like other aspects of the connectivity that aren't currently in that model that we should consider as well. I'm definitely on the same page as you. Sean?
Yeah, can I just respond briefly? I mean, Casey, I think that is one of the things that I wanted to say, that I think there are And I think Graham agrees with me. I think there are other aspects, like microstructural aspects, that will affect this. But I also just want to highlight something that somebody brought up earlier, that these are highly nonlinear systems, right? So shouldn't necessarily always expect like a linear relationship between any combination of structural measures and functional activity or connectivity measures, right? You know, we, at least when we simulate these systems, we see things like bifurcations, which can lead to strong divergences in functional activity patterns. Even when you just get like a small change in some structural measure. So I think it's at least in my head, it's a combination of, we are missing some aspects, I think, of the structure, but also that you will get, because it's a nonlinear system, I think you're guaranteed to get some divergence from the structure to the function, in terms of when you're in a nonlinear system, you shouldn't expect a linear relationship. That's how I think.
Yeah. Richard, are you following up on this, or is it?
I was going to follow up on this. I don't really like getting a little bit too down, perhaps, in the structure function debate. But maybe it also kind of depends on what you then mean by structure, because there's multiple ways that different regions of the brain can communicate, right? It's not just through axonal tracts or anything we can kind of measure with diffusion-based imaging. There's also like peptidergic signals, hormonal signals. It's not all just synaptic transmission. So if your comment is like, maybe our structural models are not comprehensive enough to capture functional connectivity, it's because we've not included every possible way that all of those regions can communicate. Do you mean that by structure, it's also those extra levels of potential communication, or is that function? I think at that level, maybe the boundary between the two becomes a little bit gray. I don't know. Not sure if that's helpful or just confusing the whole discussion, but
I think, yeah, I think that maybe it seems like we at kind of an early point in this field of like looking at structure and function. So it's nice to have these conversations about what each of us is finding we should add to these models, or how our models need to change to better understand the relationship, because I think it's like a major a question in neuroscience. So.
Absolutely. I'll leave it at that.
I completely agree with Casey. I think there's a need to develop, additional tools to visualize pathways. And that's why I was excited to look into genetics more, so that we could figure out ways to better link functional and structural connectivity, because there is something missing there. It's just how to harness other toolkits to investigate that is the question.
Yeah, maybe also to add my 2 cents before maybe I also do like a topic switch a little bit to move it a little bit away to some other elements of evolution and development, mainly the topic of this session. But like, what is really function? Because I've also done a structure function paper recently, and also another function paper. And then I was thinking, yeah, but am I looking at function, or is this just like with Casey, and like I also like, what is really like Dr. Baum, you used like an NBAC task, but also like resting state. It's rather similar, right? And I also did what you did, but then in HCP, and I found that it related to cognition, also with resting state, to how these two measures relate. But like in the end, what is then function? Can't you better do like something like, I don't know, EEG, or like a different modality, or MEG, like in a sense, where's the boundary really? What are we comparing? Isn't it not just different signals from something that is ultimately like more super granular versus like micro, or like, you know what I mean? It's this whole feature space of what we probe with these like vague measures of the brain that are awesome, but like incomplete, right? So I don't know if anybody wants to comment on that, or if you all disagree.
No, I totally agree, actually, and I think we should go multimodal, right? This is, I think, the way to tackle this kind of question. So when we're combining resting state fMRI with electrophysiology, which is something that in the mouse we can do, and in the lab we are actually currently implementing. So I hope to give you more information soon, maybe in a conference, or because we have ongoing work, which is super interesting. And yes, so multimodal to me is the answer.
Yeah, Yeah, I also, yeah. I think, and one other question what I noticed is kind of like we talked a little bit about the structural model, and then in the last talk by Graham, it was about like the structural model presupposes that regions with a similar structure have a functional relation, if I'm correct, but you presented it the other way around, that depending on its structural connectivity defines its function. Like, do you think that is, like Casey knows the exact definition of the structural model maybe better by heart, but I felt like these things are kind of different. On the one hand, speaking about a functional relationship versus a structural relationship defining the function versus the local structure between regions defining, you see what I mean? So it's like upside down. Or maybe Richard, I wrote this also to Richard, maybe he can explain this clearer, but I felt that this is like two views also on function, what it means. Like if you look at local function versus structural connection versus local structure versus functional cohesion in some way. But maybe it's too late. Maybe
I'm just generally wondering whether we need something similar for these terms as to like what Lucina did with kind of trying to get to a common jargon or a taxonomy for brain networks, or do you need something similar when we're talking about structure and function? Because to some extent, we're all talking about slightly different things, perhaps sometimes. Casey
wants to say
something. We need theories. Like that's what a theory should be. No, we have like very clearly defined theory and predictions. That then it doesn't become this like vague term, because we like to use these terms just to also have the paper read nicely as well, and we often define them at the start. But then you sometimes see that people then start relating them. And I think if we have clearly defined theories, it can help us to then move forward in that way. Yeah.
And talking about theories and evolution and development, like a lot of people like puberty or adolescence here, and like it's emerging of psychiatric disorders, but like also by Professor Jarfet, it was about birth being relevant, and also thinking about sensory regions, and like, is it really the moment that we see all these neuropsychiatric disorders emerge? Is that the moment where it happens? And is this then also related to evolution? Like a lot of big connections are made, right? But isn't it something that happens way earlier and is just like a rippling effect coming out when people start being super social with like out of family members, and then suddenly they notice something is off? Or sorry for putting this maybe a little bit stupid, but you see what I mean? Like is evolution and development, is adolescent really like the mirror imaging of what is so magic about humans in their expansion, or how do you see this? Isn't it just birth?
I definitely think about development as like a series of kind of cascading processes, where I think we've probably all touched on this, but it's really important to develop those primary sensory systems to get an accurate representation of the world and perception, reliable perception, before you can, make rapid decisions and set goals and those kinds of things, which come online much later. So I think, and so it's hard to say if you're focusing on adolescence and saying, yes, this is the time when we can, notice mental illness. I agree that you have to go back further to get a sense of how these early systems might be perturbed in a way that has this ripple effect.
Yeah, it's probably not an either or, and especially with like psychopathology or developmental conditions, we know there's a big genetic effect, and so probably a lot of that already even starts before birth. It starts at conception probably already. But then we do know that periods like adolescence and maybe other phases of development are like they have much higher variability in terms of like how that's kind of pictured across the population. So there's doubtless some interaction between some predisposition for developing that and then subsequently creating a kind of a window of opportunity for something to go wrong, perhaps. So I don't think it's either or.
I often talk about the need to identify developmental dysfunctions as early as possible, and especially how to relate information from model systems in humans, and how we need to find those corresponding time points, and how we can harness information from model systems where we can do something more invasive to really identify when abnormalities emerge really early on, and to pinpoint when that abnormality should occur in human development.
I'm thinking also about your work, starting with the visual system and like in primates, and also like looking at the gradients in mice. Like it's actually super nice how the auditory and visual system is like at this triangle, and you see that there, and the sensory motor cortex the same, right? They share like an endpoint, but it's like left, right, up. Like aren't we like a bit like, I asked this also earlier about the default mode, are we not a little bit too much hyped by its expansion, that it is super relevant, where in fact the magic is really these sensory areas that are also in the mice? Like who cares about the default mode network in the mice when you look at those pretty sensory endpoints, right? It's so nicely bordered, it's so specialized, right? And so plastic also in the studies, like seeing the posterior part of the brain, like I don't know, do you have any comments on that?
To me, you got exactly the point. So this is what I like about brain, is that they shifted the focus a bit away from this default mode network of the mouse brain by saying, okay, also primary sensory regions are super important. And in fact, they also actually cover pretty much almost 1/3, if not 2/3 of the whole cortical mantle. So they are super important, and yes, so I'm glad that somebody appreciated this aspect of the presentation. Thanks.
Yeah, no worries. Is it okay when I name people, by the way, by the first name? Because now I felt pretty bad for yes. Is this okay? Or do we say like Professor excessive? But anyway, you, Dr. or Professor Charfet, you also wanted to comment on something with the visual system, maybe, or not?
Yeah, so I think that these gradients are driven by the primary visual cortex. Rodents are just you could say maybe nearly blind, so maybe we wouldn't expect a high density of neurons in the visual cortex.
Yeah, and also thinking about disorder, like also with like Seok's the work also showing disruption in the sensory anchors, that maybe then, yeah, also what was said earlier is that it maybe is cascade, right? It starts earlier, and that without the right scaffold, you can never develop these higher order or whatever higher order or different order systems like completely, right? So maybe I'll throw the ball now to Richard to ask some questions, because I've done some turn of topic now.
I think I was just like the only other question I kind of very early on already wanted to ask, that I kind of probably cuts across all the three talks that we've seen, is the sort of translationability of all these different models, because we all talk about, and maybe even like back to Casey's talk about the default mode network, to what extent that actually represents the same thing across all these different fields that you guys study, like whether it's in mice or in primates or in humans. Is the default mode network in the same way or in the same context? Like how well does that translate across? I'm sure you all have like your own ideas about that as well. Maybe I should direct it at like maybe Ludovico, maybe like you study a mouse model specifically, so maybe you have
someone else would. Yeah, to me, this is actually an excellent question, because, for example, one aspect that it's offered that it was a bit neglected in the talk, because of course we're talking about cortical gradients, is that what we call the mouse default mode network encompasses, for example, the midline thalamus, the striatum, which, for example, in human it's not clear whether this is the case. Then for some specific brain regions, I would say that yes, there are some hints that the mouse brain could sort of host an homologue of the human default mode network. So for example, the anterior cingulate cortex as similar, which is sort of hub of the mouse default mode network, has this sort of similar cytoarchitectural properties as in humans, and etc., etc., etc. But then, of course, how does it change across evolution, and what it actually represents is the question also to me. So this is why actually I joined the Godzilla Lab to try to tackle this question more specifically.
Maybe aside from evolution, does its function change like across development, or like is that Is that something that we'd expect, or is it kind of ingrained? Maybe that's a question for Graham.
Are you asking if like the sort of like topography of the default mode changes, like in terms of which areas are fall into this network, or do you mean like the function in terms of how it's like behavioral roles?
I guess both, and I'm assuming that the two things are probably related to each other as well. If the answer to both is yes, then, but I mean that would be my suspicion, but I'm no expert in this field.
Yeah, I'm certainly not an expert in the default mode specifically, but I would expect, as far as I know, that there are shifts in the topography and kind of the spatial extent of the default mode over the course of development, from childhood through adulthood. And I would expect, I don't know research that speaks exactly to this, but I would expect things to track. I see someone just commented. I would expect things to track with, improvements in kind of like semantic and episodic memory and kind of future goal setting and those kinds of things, which I think all are just a fraction of what the default mode is involved in. But it's certainly a complicated question. I don't think I have a good answer for it.
I see Casey has her hand up, so she might have something to say about this, or a completely different topic. You're muted still, Casey.
I'm not going to go on like a default mode rant. I had like it was a kind of related follow-up question that maybe hopefully can consolidate some things that everyone's already said. Do you think that across development and evolution, there's the emergence of different gradients or extensions? So another way of saying this is, if we could describe brain organization by a low-dimensional set of gradients, kind of like the dream, do you think that if you looked at that across different animals or across development, there would be the same dimensions, but just morphed in some way, or would the dimensions actually need to be different as well? So yeah, I'll throw it to anyone can start. Graham, you don't have your mic turned off, so it's you.
That's an awesome question. Sorry, does anybody else want to start?
No, Graham, you can go.
I think that's a wonderful question. I can only speculate, but I would think it's more, I don't think like the number of dimensions change. I think it's probably just kind of a morphing within the same gradient space, if you will. And why do I think that? Why do I assume that? I would just think because there are so many similarities across species, across model systems. I think Randy Buckner and Daniel Margulies also have a paper in Marmosets showing default mode. And so I think it's highly preserved, and obviously in mice as well. And I think that I think there are common organizational principles that just are related to perception and kind of survival, but to varying degrees. Would be my guess.
I think that the gradients are evolving based on the developmental processes that are changing. So in one case, you have variation in the duration of neurogenesis that gives rise to variation in cytoarchitecture, but there can be other kinds of developmental processes evolving in different mammalian taxonomic groups. So one might imagine the growth of pathways, there's maybe a heterochrony there in some mammalian species. So I guess the way maybe to define those gradients is based on the developmental processes that are evolving.
And I actually agree totally with Graham, because so what I just would add on top of this, sort of anecdotally, is that, for example, in the mouse brain, we think that the primary visual cortex is part of the default mode network. There is strong functional and structural connectivity. And so what happens probably during evolution, also because the primary visual cortex in the mouse brain has actually responds quite strongly also to auditory stimuli. So to what extent it's really unimodal is under debate. And so what I think I would speculate that happens is also sort of we have this morphing in the same gradient space in which, for example, then the primary visual cortex detaches from the default mode network in higher species.
And maybe related, we also had maybe a question from Bing in the chat about if there's no visual input from birth, do you expect the same high neuron density at the visual cortex? And maybe to broaden up this question a little bit, like, is it nature or nurture, or like, is it our genetic makeup that shapes our gradients, or is it also that we live our lives that in the end builds the tent that is the gradient space in our brains? Or like maybe first one person can answer specifically this question and then like speculate on the second part.
I think there was a study looking at neuron densities in a newborn primate, and I believe they found a gradient. It's from my memory. And so I think that the gradient emerges prenatally and can then be sculpted postnatally.
Yeah, I would also agree that I think also like broader related to this, like how like is there like a finite number of gradients, because we have like this posterior anterior gradient that kind of relates to this like Daniel's gradient, so to say, from like unimodal or ideotypic to default mode, but we have also this sensory-fugal gradient, and it's not exactly the same, at least in my opinion, like seeing these shifts in myelination from granular to agranular, and like dual origin is also like this organization principle that kind of fits but doesn't fit that nicely in humans. So like, where does it end, right? Like, things do correlate, and we do the spin test and we show, okay, well, there's 0.4 correlation, but that's obviously not perfect. So what does it then mean, you know? I love having a correlation that is significant, you know? I'm not lying there, but in the end, what does it mean when two things relate to each other? Is it then the same, or is it just different elements of the same phenomena, or actually something completely different that we're describing, relating to layers or relating to the state that the person is in, or how do you guys see this?
Can I just quickly echo what Christine said before about biological processes being a really good way to define a finite number, possibly, rather than looking at like the phenomena in the adult brain? We could define these dimensions better by looking at biological processes earlier instead, and then by their mergence and like crisscrossing and intersection would understand all the phenomena that we're looking at now.
But maybe as somebody that did a PhD in social neuroscience, maybe to put this back to you, like, what does this then mean? Like, for example, thinking about the gradient 3, right? Like this dissociation between like external, like attention-oriented cognition versus more internally thought. Like how does this then relate to psychology? Because some of us, including me, sometimes try to understand behavior through gradients, and that's like far away from transcriptome, right? So can we really bridge all these skills with one gradient, or how do you then see that?
I can try to feel this, but you know I don't touch behavior, right?
Well, you do behave, right? So
I do, but yeah, that's true. Well, I guess it means, I think with the default mode, like by looking at just a couple of cytoarchitectural gradients, I feel like I have a much better understanding. It was like the first time I feel like I've ever touched cognitive neuroscience. And just by understanding its connectivity and its cytoarchitecture, I now have a better idea of how the default mode could be reconstructing information, how it helps us act in the world. And that was just like 2 cytoarchitectural gradients, actually. So I think that the potential's there. I don't think it's been actualized yet, though. So I think it's too early to say whether, if we use this low dimensional set of gradients, we can explain behavior. I see promise, but I don't see it fully explored yet.
And how do other people see this? Are they?
Well, there's the inclusion of the model system where you can do, manipulations and see how to tie those, biological and behavioral processes you're interested in, and make sure that they're connected in the model system.
And maybe also like Graham, based on like your correlation also with executive function and like linking this to, how do you see this? do you think that one can combine it all? Like you have a map of evolution and individual differences. Can we really go that far with one gradient?
I think, yeah, I think it's a useful, the gradient's a useful interpretive lens. I agree that, yeah, at what point do you pose the question of like, when do we stop? You know, you can find a map like the dual origin map or the laminar organization and all these things, which are probably related to a lot of the things that we're interested in because they are ways that the brain is organized. But I think I agree that it becomes challenging to determine kind of what are the real. I've only thought about this after I had already submitted this paper, but I was thinking, so if like if I find results in like the dorsolateral prefrontal cortex for anything, does that mean that my results are always related to evolutionary expansion? Like, is this even a meaningful association? And so I have since.
Yes.
Right.
Yes, I'll just help you.
You think so?
No, I don't know, but I wasn't.
But yeah, so it's definitely a challenging thing. And I think a lot of it comes back to kind of when we were talking about the structure function stuff, a lot of it's like about the units, like the spatiotemporal units, I guess, like how precise we can get. And what these gradients, which are kind of like broad organizational principles, if they're granular enough to predict behavior. That's like one of the reasons that I decided to look at the level of individual like cortical areas rather than just deriving a structural gradient and a functional gradient, because we wanted to see if there were more nuanced differences within networks or at a smaller spatial scale. But I imagine in model systems, you can go even smaller. And, you know, I think there's actually some work like Rick Betzel, a friend of mine, and who many of you I'm sure know, has published a paper looking at ECOG data and its relationship to genetic and structural connectivity. And possibly, I'm not sure if he has behavior in there, but I think like the more spatial and temporal scales we can incorporate into this model, the better chance we'll have at understanding behavior, to summarize.
Yes, I definitely agree with Graham. So, and the mice offered this opportunity because we could combine, for example, optogenetics, so with behavior and simultaneous neural recording also across multiple spatial scales, and that would be actually a super nice experiment to perform to see whether gradients can explain something in behavior, for example.
I think in addition to the mouse model you just discussed is looking at convergence, looking to see whether the the biology and behavior has evolved in different taxonomic groups. If you see similar patterns of evolution in distantly related species, that also adds to this idea that the biological change and the behavioral change are causally linked.
Yeah, I thank you for that. I think there are many different ways to, at least I've used gradients in different ways and thought about them differently over the last years, but I think many of us have done this in different ways. I think it's also now almost time for people to go to Wonder Me. So maybe Casey or Boris, I don't know who's the pilot here. Or stewardess.
Yeah, come on.
Boris has to laugh. Sorry, it's an inside joke. But...
Okay, yeah, thank you. I love talking about development evolution. And it's quite funny that we, as a field, somehow all converge upon structure and function. I didn't realize that was going to come out so naturally from all of us talking about development evolution. So that was really cool. Okay, so that's all for today. Thank you so much to the speakers of this session. Thank you, Sophie and Richard, for leading us through. That's all of the talk content for today. But what I'll do is, and also I should thank once again, let me just share my screen for a second. I just wanted to have another opportunity to thank our sponsors, because this is really important that to make this whole workshop work well. So they have been really kind in supporting us this year, even as we go virtual, but there's still a lot of organization behind this. And there we go. So thank you again to all our sponsors for making this possible. So ULISH, Human Brain Project, Highball, AirBIC, and the Neuro. And also thank you so much to the Neuro events organizing team, because there's a lot that happens behind the scenes, especially today, and they've been really, really helpful the whole way. And hopefully made it a really worthwhile experience for everyone attending. I think at the maximum, I saw like 150 people on the Zoom call. That's awesome. Okay, so with that, we can move over to Wanda. So I think either I or someone will drop the link in the chat in a second, and that's where you can find the poster rooms and just like places to gather and chat. You can either just meet up with people like you can organize with, but also there's poster rooms that are clustered by themes. So you can just go into one of these themed rooms, and people who have posters to present will just take turns sharing their screen and talking through their work. And then there's also a couple of larger rooms that I've created if you just want to go into, and we can continue these type of conversations. There's also a football match starting in about an hour that if people are still hanging around, I'm going to screen the Italy versus Switzerland match in Wanda as well, if we're still talking then, and we can just, yeah, I'm opening a beer immediately as well. So I'll see you over on Wanda, but thank you again for all our speakers, chairs, organizers, sponsors, and all the attendees for making this a really fun day. Yeah, and if there's nothing else, I'll see you all over on Wanda.
Thank you.
Thank you, Casey, and all the speakers.
Thanks.
Yeah, thanks everyone. Casey, that was great. So really great day.
Yeah, I'm just making sure no one has any questions, but you guys can all head off over to the poster session, because I think there's some nice, there's also details of the posters in the program booklet if you want to check out first what everything is. Everyone's filtering out. Okay, I think we're good. I'm going to head over to Wanda too.